Chapter 3 Recombinant DNA Technology and Genomics. Undergraduate biology students working on a recombinant DNA experiment. After completing this chapter, you should be able to define recombinant DNA technology and explain how it is used to clone genes and manipulate DNA. Compare and contrast different types of cloning vectors, describe their practical features and applications. Discuss how DNA libraries are created and screened to identify cloned genes of interest. Describe how agarose gel electrophoresis, polymerase chain reaction, PCR, DNA sequencing, and other molecular techniques are used to study gene structure, function, and expression. Describe how whole genome shotgun sequencing and high-throughput sequencing techniques enable scientists to rapidly analyze genomes. Understand major findings of the Human Genome Project, potential scientific and medical applications of knowledge about the human genome, and associated ethical, legal, and social issues. Explain why genomics-related omics disciplines are developing rapidly as areas of research. Provide examples of how bioinformatics can be used to analyze nucleic acid and protein sequences and structures. As you have learned, biotechnology is not a new science. However, the modern era of biotechnology began when DNA cloning techniques were developed. Since the 1970s and continuing today, amazing and rapidly developing laboratory methods in recombinant DNA technology and genetic engineering have changed molecular biology, basic science, and medical research forever. In this chapter, we present an overview of recombinant DNA technology. We then take a look at an amazing range of techniques that scientists use to clone genes and study gene structure and function. The chapter concludes with an introduction to genomics and bioinformatics, which provide methods for studying genomes. Forecasting the future. There are many exciting potential future directions in recombinant DNA and genomics research. Without question, one area of substantial progress will be the completion of thousands of genome projects for different plants, animals, bacteria, viruses, and other organisms. Scientists continue to prospect genomes from organisms around the world to look for new genes with potential applications in biotechnology. Following on the success of the Human Genome Project, the use of genomic information for new applications, including novel methods for the early detection of disease genes and novel gene-based treatment strategies for diseases such as cancer and many other human health conditions, holds tremendous promise for alleviating pain and suffering through biotechnology. And on the near horizon is the sequencing, personal genomics, of individual genomes. 3.1 Introduction to Recombinant DNA Technology and DNA Cloning. When scientists James Watson and Francis Crick discovered that the structure of DNA is a double helical molecule, they hinted at the potential importance and impact of this discovery. However, not even these Nobel Prize winners could have imagined the astonishing pace at which molecular biology would advance over the next half-century. In the years before and after Watson and Crick's discovery, many other scientists contributed to our understanding of DNA as the genetic material of living cells. A number of researchers studied DNA structure and replication in bacteria and in bacteriophages. Bacteriophages, often simply called phages, are viruses that infect bacterial cells. Much of what we know about DNA replication and DNA synthesizing enzymes has been learned from studying bacteria and phages. For example, recall that DNA ligase joins together adjacent DNA fragments, Okazaki fragments, during DNA replication, Chapter 2. As you will soon learn, DNA ligase is an important enzyme in recombinant DNA technology. In the late 1960s, many scientists were interested in gene cloning. They speculated that it might be possible to clone DNA by cutting and pasting DNA from different sources, recombinant DNA technology. It may seem that the terms gene cloning, recombinant DNA technology, and genetic engineering describe the same process. Recombinant DNA technology is commonly used to make gene cloning possible, whereas genetic engineering often relies on recombinant DNA technology and gene cloning to modify an organism's genome. However, the terms recombinant DNA technology and genetic engineering are frequently used interchangeably. Clone is derived from a Greek word that describes a cutting of a twig that is used to propagate or copy a plant. A modern biological definition of a clone is a molecule, cell, or organism produced from another single entity. The laboratory methods required for gene cloning as described in this chapter are different from the techniques used to clone whole organisms. Chapter 7. Restriction enzymes and plasmid DNA vectors. In the early 1970s, gene cloning became a reality. Many nearly simultaneous discoveries and collaborative efforts among several researchers led to the discovery of two essential components that made gene cloning and recombinant DNA techniques possible: restriction enzymes and plasmids. Plasmid DNA. Restriction enzymes are DNA cutting enzymes, and plasmid DNA is a circular form of self-replicating DNA that scientists can manipulate to carry and clone other pieces of DNA. Microbiologists in the 1960s discovered that some bacteria are protected from destruction by bacteriophages because they can restrict phage replication. Swiss scientist Werner Arber proposed that restricted growth of phages occurred because some bacteria contained enzymes that could cut viral DNA into small pieces, thus preventing viral replication. Because of this ability, these enzymes were called restriction enzymes. In 1970, working with the bacterium Haemophilus influenza, Johns Hopkins University researcher Hamilton Smith isolated Hind III, the first restriction enzyme to be well characterized and used for DNA cloning. Restriction enzymes are also called restriction endonucleases, endo, within, nuclease, nucleic acid cutting enzyme, because they cut within DNA sequences as opposed to enzymes that cut from the ends of DNA sequences, exonucleases. Smith demonstrated that Hind III could be used to cut or digest DNA into small fragments. In 1978, Smith shared a Nobel Prize with Werner Arber and Daniel Nathans for their discoveries on restriction enzymes and their applications. Restriction enzymes are primarily found in bacteria, and they are given abbreviated names based on the genus and species names of the bacteria from which they are isolated. For example, one of the first restriction enzymes to be isolated, EcoRI, is so named because it was discovered in the E. coli strain called RY13. Restriction enzymes cut DNA by cleaving the phosphodiester bond, in the sugar phosphate backbone, that joins adjacent nucleotides in a DNA strand. However, restriction enzymes do not just randomly cut DNA, nor do all restriction enzymes cut DNA at the same locations. Like other enzymes, restriction enzymes show specificity for certain substrates. For these enzymes, the substrate is DNA. As shown in Figure 3.1a, restriction enzymes bind to, recognize, and cut, digest, DNA within specific sequences of bases called restriction sites. Why don't restriction enzymes digest DNA in bacterial cells? Bacteria protect their DNA from restriction enzyme digestion because some of the nucleotides in their DNA contain methyl groups that block restriction enzymes from digestion. Figure 3.1b. Restriction enzymes are commonly referred to as 4 or 6 base pair cutters because they typically recognize restriction sites with a sequence of 4 or 6 nucleotides. 8 base pair cutters have also been identified. Each restriction site is a palindrome. The arrangement of nucleotides reads the same forward and backward on opposite strands of the DNA molecule. Remember the word madam or the phrase a Toyota as examples of palindromes. Some restriction enzymes, such as EcoRI, cut DNA to create DNA fragments with overhanging single-stranded ends called sticky or cohesive ends. See Figure 3.1a. Other enzymes generate fragments with double-stranded ends called blunt ends. Table 3.1 shows some common restriction enzymes, their source microorganisms, and their restriction sites. Notice that the first 3 enzymes in the table are 6 base pair cutters that produce DNA molecules with cohesive ends. The fourth enzyme, TCI, is a 4 base pair cutter that produces cohesive ends, and the lower 3 enzymes produce blunt-ended DNA fragments. Enzymes that produce cohesive ends are often Figure 3.1 Restriction sites and restriction enzyme action. A. Digestion of DNA by EcoRI produces DNA fragments with cohesive ends. B. Methylation of the EcoRI restriction site by the enzyme EcoRI methylase blocks DNA cleavage by EcoRI. Note: methylation of the EcoRI restriction site occurs on an adenine nucleotide, but methylation more frequently occurs on cytosine nucleotides. Table 3.1 Common restriction enzymes. Favored over blunt end cutters for many cloning experiments because DNA fragments with cohesive ends can easily be joined together. DNA from any source such as bacteria, humans, dogs, cats, frogs, dinosaurs, or ancient human remains can be digested by a particular restriction enzyme as long as the DNA has a restriction site for that enzyme. In the simplest sense, the discovery of restriction enzymes provided molecular biologists with the scissors needed to carry out gene cloning. Tools of the trade: restriction enzymes. Restriction enzymes are sophisticated scissors that molecular biologists use to manipulate DNA. Working with restriction enzymes has become easier over the 30 years, since Hamilton Smith and others pioneered their use. Over 300 restriction enzymes are commercially available rather inexpensively. Many enzymes are readily available because they have been cloned using recombinant DNA technology, so they are made and isolated in large quantities. Commercially prepared enzymes come in conveniently sized prepackages with buffer solutions that provide all the components necessary for optimal enzyme activity. If researchers must work with an enzyme with which they are unfamiliar, they can use the restriction enzyme database Rebase, an outstanding tool for locating enzyme suppliers and enzyme specifics. In addition, a variety of software packages and websites are available to assist scientists who work with restriction enzymes and DNA sequences. For example, imagine that you are a molecular biologist who just cloned and sequenced a 7,200 BP piece of DNA and you want to see if there is an enzyme that will cut your gene to create a 250 BP piece of DNA for a probe you want to make. Not too long ago, if you had a lot of enzymes in your freezer, you could digest this DNA and run gels to see if you could get a 250 BP piece, but this imprecise approach took a lot of time and resources. If you had sequenced your gene, you could scan the sequence with your eyes, looking for a restriction site of interest, a very time-consuming and eye-straining effort. The internet makes this task much easier, because many websites function as online tools for analyzing restriction enzyme cutting sites. For example, in WebCutter, DNA sequences can be entered and searched to determine restriction enzyme cutting patterns. See Problem 8 in Questions and Activities. The widespread application of recombinant DNA techniques in many areas of biological and medical research has led to hundreds of technique books, websites, and journals. In Biotechniques, a popular monthly journal, biologists publish and share information on molecular cloning techniques. Several sites that are commonly used for designing PCR primers and other applications are provided in Keeping Current, web links, on the companion website. In the early 1970s, Paul Berg, Herbert Boyer, Stanley Cohen, and colleagues at Stanford University used gene cloning to change molecular biology forever. Berg founded recombinant DNA technology when he created a piece of recombinant DNA by joining together, splicing, DNA from the E. coli chromosome and DNA from a primate virus called SV40, simian virus 40. Berg first isolated chromosomal DNA from E. coli and DNA from SV40. Then he cut both DNA samples with EcoRI, added E. coli DNA and viral DNA fragments to a reaction tube with the enzyme DNA ligase, and succeeded in creating a hybrid molecule of SV40 and E. coli DNA. The importance of this discovery was fully recognized when Paul Berg won the 1980 Nobel Prize in Chemistry for this experiment, which demonstrated that DNA could be cut from different sources with the same enzyme and that the restriction fragments could be joined to create a recombinant DNA molecule. Berg shared this prize with Walter Gilbert and Frederick Sanger, who independently developed methods for sequencing DNA, a process we discuss later in this chapter. Berg worked with chromosomal DNA, Cohen was interested in the molecular biology of small circular pieces of DNA known as plasmids. Plasmid DNA is primarily found in bacteria. Plasmids are primarily found, and are considered extrachromosomal DNA because they are present in the bacterial cytoplasm in addition to the bacterial chromosome. Plasmids are small, most average approximately 1,000 to 4,000 base pairs, BP, in size, and they are self-replicating, that is, they duplicate independently of the chromosome. Cohen studied plasmid replication, the transfer of plasmids between bacteria, and mechanisms of bacterial resistance to antibiotics. Cohen postulated that plasmids could be used as vectors, pieces of DNA that can accept, carry, and replicate, clone, other pieces of DNA. Cohen and Boyer worked with 2 bacterial plasmids to clone DNA successfully. They published results describing these experiments in 1973. Many consider this work the informal birth of recombinant DNA technology. Using EcoRI, a restriction enzyme previously isolated by Herbert Boyer, they cut both plasmids and then joined fragments from each plasmid together using DNA ligase to create new hybrid, recombinant, plasmids. Recall that DNA ligase catalyzes the formation of phosphodiester bonds between nucleotides. Ligase can join together DNA with cohesive ends as well as blunt-ended fragments. As a result of these and other experiments, Cohen and Boyer produced the first plasmid vector for cloning purposes, called PSC101 and named SC for Stanley Cohen. PSC101 contained a gene for tetracycline resistance and restriction sites for several enzymes including EcoRI and HindIII. In subsequent work they used similar experiments to clone DNA from the South African claw-toed frog Xenopus laevis, another important model organism in genetics and developmental biology, into the EcoRI site of PSC101. Figure 3.2 illustrates how recombinant DNA can be formed in a process similar to that used in the Cohen and Boyer experiments. Figure 3.2 Creating recombinant DNA. EcoRI binds to a specific sequence, 5-GOT-C3, and then cleaves the DNA backbone, producing DNA fragments. The single-stranded ends of the DNA fragments can form hydrogen bonds with each other because they have complementary base pairs. DNA ligase can then catalyze the formation of covalent bonds in the DNA backbones of the fragments to create a piece of recombinant DNA. Cohen and Boyer had created the first DNA cloning vector, a vehicle for the insertion and replication of DNA, and in 1980 were awarded patents for PSC101 and for the gene splicing and cloning techniques they had developed. These experiments ushered in the birth of modern biotechnology, because many of the current techniques used for gene cloning and gene manipulations are based on these fundamental methods of recombinant DNA technology. In 1974, as a direct result of the Berg, Cohen, and Boyer experiments, gene cloning pioneers and critics voiced concerns about the safety of genetically modified organisms. Scientists were concerned about what might happen if recombinant bacteria were to leave the lab, or if such bacteria could transfer their genes to other cells or survive in other organisms, including humans. In 1975, an invited group of well-known molecular biologists, virologists, microbiologists, lawyers, and journalists gathered at the Asilomar Conference Center in Pacific Grove, California, to discuss the benefits and potential hazards of recombinant DNA technology. As a result of the historic Asilomar meeting, the National Institutes of Health, NIH, formed the Recombinant DNA Advisory Committee, RAC, which was charged with evaluating the risks of recombinant DNA technology and establishing guidelines for recombinant DNA research. In 1976, the RAC published a set of guidelines for working with recombinant organisms. The RAC continues to oversee gene cloning research, and compliance with RAC guidelines is mandatory for scientists working with recombinant organisms. Transformation of bacterial cells and antibiotic selection of recombinant bacteria. Cohen also made another important contribution to gene cloning, which made the PSC101 cloning experiments possible. His laboratory demonstrated how transformation, a process for inserting foreign DNA into bacteria, could be used to reliably introduce DNA into bacteria. Cohen discovered that if he treated bacterial cells with calcium chloride solutions, added plasmids to cells chilled on ice, and then briefly heated the cell and DNA mixture, plasmids entered bacterial cells. Once inside bacteria, plasmids replicate and express their genes. Transformation techniques are explained in greater detail later, Chapter 5. A more modern transformation method, called electroporation, involves applying a brief, millisecond, pulse of high-voltage electricity to create tiny holes in the bacterial cell wall that allow DNA to enter. Electroporation can also be used to introduce DNA into mammalian cells and to transform plant cells. Ligation of DNA fragments and transformation by any method are somewhat inefficient. During ligation, some of the digested plasmid ligates back to itself to create recircularized plasmid that lacks foreign DNA. During transformation, a majority of cells do not take up DNA. Now that you have learned how DNA can be inserted into a vector and introduced into bacterial cells, we consider how recombinant bacteria, those transformed with a recombinant plasmid, can be distinguished from a large number of non-transformed bacteria and bacterial cells that contain plasmid DNA without foreign DNA. This screening process is called selection because it is designed to facilitate the identification of, selecting for, recombinant bacteria while preventing the growth of, selecting against, non-transformed bacteria and bacteria that contain plasmid without foreign DNA. Cohen and Boyer used antibiotic selection, a technique in which transformed bacterial cells are plated on agar plates with different antibiotics, as a way to identify recombinant bacteria and non-transformed cells. For many years, antibiotic selection was a widely used approach. Modern cloning techniques often incorporate other more popular selection strategies, such as blue-white selection. The reason for this name will soon be obvious. In blue-white selection, DNA is cloned into a restriction site in the LAX gene, as illustrated in Figure 3.3. Recall that the LAX gene encodes beta-galactosidase, beta-gal, an enzyme that degrades the disaccharide lactose into the monosaccharides glucose and galactose, Chapter 2. When it is interrupted by an inserted gene, the LAX gene is incapable of producing functional beta-gal. Transformed bacteria are plated on agar plates that contain an antibiotic ampicillin, in this example. Non-transformed bacteria cannot grow in the presence of ampicillin because they lack plasmids containing an ampicillin resistance gene, AMPR. But antibiotic selection alone does not distinguish transformed bacteria with non-recombinant plasmid that has recircularized from recombinant plasmids. To identify bacteria with recombinant plasmids, the agar must also contain a chromogenic, color-producing, substrate for beta-gal called X-gal, 5-bromo-4-chloro-3-indolyl beta-D-galactopyranose. X-gal is similar to lactose in structure and turns blue when cleaved by beta-gal. As a result, non-recombinant bacteria, those that contain plasmid that ligated back to itself without insert DNA, contain a functional LAX gene, produce beta-gal, and turn blue. Conversely, recombinant bacteria are identified as white colonies. Because these cells contain plasmid with foreign DNA inserted into the LAX gene, beta-gal is not produced, and these cells cannot metabolize X-gal. Therefore, through blue-white selection. Figure 3.3, cloning a gene in a plasmid and blue-white selection. Non-transformed and non-recombinant bacteria are selected against and white colonies are identified or selected for as the desired colonies containing recombinant plasmids. Colonies containing recombinant plasmids are clones, genetically identical bacterial cells each containing copies of the recombinant plasmids. Introduction to human gene cloning. Restriction enzymes, DNA ligase, and plasmids are major tools of molecular biologists for manipulating and cloning genes from virtually any source. With transformation, scientists had a way to introduce recombinant DNA into bacterial cells. Recombinant DNA technology made it possible to cut and join together DNA fragments, insert DNA into a plasmid. DNA cloned into a plasmid is commonly called insert DNA, and produce large amounts of the insert DNA by allowing bacteria to be the workhorses for replicating recombinant DNA. If the cloned DNA fragment is a gene that encodes a protein product, bacterial cells could be used to synthesize the protein product of the cloned gene. We call this expressing a protein. Molecular biologists recognized that if human genes could be cloned and expressed, recombinant DNA technology would become an invaluable tool with powerful and exciting applications in research and medicine. Because bacteria can be grown in large-scale preparations, scientists can produce large amounts of the cloned DNA and isolate quantities of protein that would normally be very difficult or expensive to purify without cloning. These processes are described in more detail in chapters 4 and 5. Because of recombinant DNA technology, a wide range of valuable proteins that are otherwise difficult to obtain can be produced from cloned genes. The first commercially available human gene product of recombinant DNA technology was human insulin, a peptide hormone produced by cells in the pancreas called beta cells. When blood glucose rises, for example, after eating a sugar-rich meal, insulin lowers blood glucose by stimulating glucose storage in liver and muscle cells as long chains of glucose called glycogen. Individuals with type I, or insulin-dependent, diabetes mellitus do not produce insulin on their own. As a result of this insulin deficiency, diabetics experience excessively high blood sugar levels, hyperglycemia, which, over time, can lead to serious damage to many bodily organs. In 1977, the insulin gene was cloned into plasmids, expressed in bacterial cells, and isolated by scientists at Genentech, named for genetic engineering technology, the San Francisco biotechnology company co-founded in 1976 by Herbert Boyer and Robert Swanson. Details of the techniques used for cloning insulin are examined in Chapter 5. Genentech is generally regarded as the first biotechnology company. In 1982, the recombinant form of human insulin, called Humulin, became the first recombinant DNA product to be approved for human applications by the U.S. Food and Drug Administration, FDA. Shortly after insulin became available, growth hormone used to treat children who suffer from a form of dwarfism was cloned, and because of recombinant DNA technology, a wide variety of other medically important proteins that were once difficult to obtain in adequate amounts became readily available. Prior to recombinant DNA technology, important hormones like insulin and growth hormone had to be isolated from tissues. Growth hormone was isolated from the pituitary glands of human cadavers. Not only was this process expensive and inefficient, but these isolations also carried with them the risk of unknowingly CO-purifying viruses and other pathogens as contaminants that could be passed to people receiving the hormone. There are now several hundred products of recombinant DNA technology on the market with widespread applications in basic research, medicine, and agriculture. With a basic understanding of the techniques involved in manipulating a piece of DNA, in the next section we go on to examine some important aspects of DNA vectors and how different vectors are chosen and used depending on what is to be accomplished. 3.2 What makes a good vector? The number of different DNA vectors, vector functions, and applications has increased substantially since Stanley Cohen constructed PSC-101. Plasmids are still the most commonly used cloning vectors because they allow for the routine cloning and manipulation of small pieces of DNA that form the foundation for many techniques used daily in a molecular biology laboratory. In addition, it is fairly simple to transform bacterial cells with plasmids and relatively easy to isolate plasmids from bacterial cells. One of the first widely used plasmid vectors, called PBR322, was designed to include genes for ampicillin and tetracycline resistance and several useful restriction sites. However, plasmid cloning vectors have been engineered over the years to incorporate a number of other important features that have made PBR322 obsolete. Practical features of DNA cloning vectors. Plasmid cloning vectors usually include most of the following desirable and practical features. Size: they should be small enough to be easily separated from the chromosomal DNA of the host bacteria. Origin of replication, ORI. site for DNA replication that allows plasmids to replicate separately from the host cell's chromosome. The number of plasmids in a cell is called copy number. The normal copy number of plasmids in most bacterial cells is small, usually less than 12 plasmids per cell. However, many of the most desirable cloning plasmids are known as high copy number plasmids because they replicate to create hundreds or thousands of plasmid copies per cell. Multiple cloning site, MCS. The MCS is a segment of DNA with recognition sites for different common restriction enzymes. See Figure 3.3a. These sites are engineered into the plasmid so that digestion of the plasmid with restriction enzymes does not result in the loss of a fragment of DNA. Rather, the circular plasmid simply becomes linearized when it is digested with a restriction enzyme. An MCS provides for great flexibility in the range of DNA fragments that can be cloned into a plasmid because it is possible to insert DNA fragments generated by cutting with many different enzymes. Selectable marker genes. These genes allow for the selection and identification of bacteria that have been transformed with a recombinant plasmid. Some of the most common selectable markers are genes for ampicillin resistance, AMPR, tetracycline resistance, TETR, and the LAX gene used for blue-white selection. RNA polymerase promoter sequences. These sequences are used for the transcription of RNA in vivo and in vitro by RNA polymerase. Recall that RNA polymerase copies DNA into RNA during transcription, Chapter 2. In vivo, these sequences allow bacterial cells to make RNA from cloned genes, which in turn leads to protein synthesis. In vitro, transcribed RNA can synthesize RNA probes, which are useful in studying gene expression, Section 3.4. DNA sequencing. Primer sequences. These sequences permit nucleotide sequencing of cloned DNA fragments that have been inserted into the plasmid, Section 3.4. Types of vectors. Just as one screwdriver cannot be used for all sizes and types of screws, bacterial plasmid vectors cannot be used for all applications in biotechnology. There are limitations to how plasmids can be used in cloning. One primary limitation is the size of the DNA fragment that can be inserted into a plasmid. Insert size usually cannot exceed approximately 6 to 7 kilobases, 1 kb equals 1000 bp. In addition, sometimes bacteria express proteins from eukaryotic genes poorly. Because of these limitations, molecular biologists have developed many other types of DNA vectors, each of which has particular benefits depending on the cloning application. Table 3.2 compares the important features, sources, and applications of different types of cloning vectors. Table 3.2 A comparison of DNA vectors and their applications. Vector type. Maximum insert size, kb. Applications. Limitations. Bacterial plasmid vectors, circular. 612. DNA cloning, protein expression, subcloning, direct sequencing of insert DNA. Restricted insert size, limited expression of proteins, copy number problems, replication restricted to bacteria. Bacteriophage vectors, linear. 25. cDNA, genomic and expression libraries. Packaging limits DNA insert size, host replication problems. Cosmid, circular. 35. cDNA and genomic libraries, cloning large DNA fragments. Phage packaging restrictions, not ideal for protein expression, cannot be replicated in mammalian cells. Bacterial artificial chromosome, BAC, circular. 300. Genomic libraries, cloning large DNA fragments. Replication restricted to bacteria, cannot be used for protein expression. Yeast artificial chromosome, YAC, circular. 202,000. Genomic libraries, Cloning large DNA fragments. Must be grown in yeast, cannot be used in bacteria. TI vector, circular. Varies depending on type of TI vector used. Gene transfer in plants. Limited to use in plant cells only, number of restriction sites randomly distributed, large size of vector not easily manipulated. Bacteriophage vectors. DNA from bacteriophage lambda, was one of the first phage vectors used for cloning. The lambda chromosome is a linear structure approximately 49 kb in size. Cloned DNA is inserted into restriction sites in the center of the lambda chromosome. Recombinant chromosomes are then packaged into viral particles in vitro, and these phages are used to infect E. coli growing as a lawn, a continuous layer covering the plate. At each end of the lambda chromosome are 12 nucleotide sequences called cohesive sites, COS, which can base pair with each other. When lambda infects E. coli as a host, the lambda chromosome uses COS sites to circularize and then replicate. Bacteriophage lambda L replicates through a process known as a lytic cycle. As lambda replicates to create more viral particles, infected E. coli are lysed. To lyse means to split or rupture, by lambda, creating zones of dead bacteria called plagues, which appear as cleared spots on the bacterial lawn. Each plaque contains millions of recombinant phage particles. An advantage of these vectors is that they allow for the cloning of larger DNA fragments, up to approximately 25 kb, than plasmids. Phage vectors are no longer very widely used. Cosmid vectors. Cosmid vectors contain COS ends of lambda DNA, a plasmid origin of replication, and genes for antibiotic resistance, but most of the viral genes have been removed. DNA is cloned into a restriction site and the cosmid is packaged into viral particles, as is done with bacteriophage vectors, and used to infect E. coli, wherein cosmids replicate as a low copy number plasmid. Bacterial colonies are formed on a plate, and recombinants can be screened by antibiotic selection. One advantage of cosmids is that they allow for the cloning of DNA fragments in the 20 to 45 kb range. But much like phage vectors, because of the development of other types of vectors, cosmid vectors are no longer very widely used and have limited applications. Expression vectors. Protein expression vectors allow for the high-level synthesis, expression, of eukaryotic proteins within bacterial cells because they contain a prokaryotic promoter sequence adjacent to the site where DNA is inserted into the plasmid. Bacterial RNA polymerase can bind to the promoter and synthesize large amounts of RNA, for the insert, which is then translated into protein. Protein may then be isolated using biochemical techniques, described in Chapter 4. However, it is not always possible to express a functional protein in bacteria. For example, bacterial ribosomes sometimes cannot translate eukaryotic mRNA sequences. If a protein is produced, it may not fold and be processed correctly, as occurs in eukaryotic cells that use organelles to fold and modify proteins. Also, making some recombinant products in bacteria can be a problem because E. coli often does not secrete proteins. Therefore expression vectors are often used in Bacillus subtilis, a strain more suitable for protein secretion. In some cases, the host bacteria can recognize recombinant proteins as foreign and degrade the protein, whereas in others the expressed protein is lethal to the host bacterial cells. Certain viruses, such as SV40, can be used to deliver expression vectors into mammalian cells. Typically SV40-derived vectors contain a strong, viral, promoter sequence for high-level transcription in a poly-A addition signal for adding a poly-A tail to the 3 end of synthesized mRNAs. Variations of such vectors have been used for human gene therapy. Bacterial artificial chromosomes. Bacterial artificial chromosomes, BACs, are large low-copy number plasmids present as 1 to 2 copies in bacterial cells and they containing genes that encode the F factor, a unit of genes controlling bacterial replication. BACs can accept DNA inserts in the 100 to 300 kb range. BACs were widely used in the Human Genome Project to clone and sequence large pieces of human chromosomes. Yeast artificial chromosomes. Yeast artificial chromosomes, YACs, are small plasmids grown in E. coli and introduced into yeast cells, such as Saccharomyces cerevisiae. A YAC is a miniature version of a eukaryotic chromosome. YACs contain an origin of replication, selectable markers, 2 telomeres, and a centromere that allows for replication of the YAC and segregation into daughter cells during cell division. Foreign DNA fragments are cloned into a restriction site in the center of the YAC. YACs are particularly useful for cloning large fragments of DNA from 200 kb to approximately 2 megabases, MB equals 1 million bases, in size. Like BACs, YACs played an important role in the cloning efforts of the Human Genome Project. TI vectors. TI vectors are naturally occurring plasmids, around 200 kb in size, isolated from the bacterium Rhizobium radiobacter, formerly called Agrobacterium tumefaciens and recently renamed on the basis of genome data, a soil-borne plant pathogen that causes a condition in plants called crown gall disease. When our radiobacter enters host plants, a piece of DNA, tDNA, from the TI plasmid, TI stands for tumor-inducing, inserts into the host chromosome. tDNA encodes for the synthesis of a hormone called auxin, which weakens the host's cell wall. Infected plant cells divide and enlarge to form a tumor, gall. Plant geneticists recognized that if they could remove auxin and other detrimental genes from the TI plasmid, the resulting vector could be used to deliver genes into plant cells. TI vectors are widely used to transfer genes into plants, as you will learn in Chapter 6. Now that we have examined different types of vectors and their applications, in the next section we will turn our attention to how scientists can use recombinant DNA technology to identify and clone genes of interest. 3.3 How do you identify and clone a gene of interest? Cutting and pasting different pieces of DNA to produce a recombinant DNA molecule has become a routine technique in molecular biology. But the types of cloning experiments we have described so far allow for the random cloning of DNA fragments based on restriction enzyme cutting sites, not precise cloning of a single gene or particular piece of DNA of interest. For example, if you were interested in cloning the insulin gene and you simply took DNA from the pancreas, cut it with enzymes, and then ligated digested DNA into plasmids, you would create hundreds of thousands of recombinant plasmids, and not just recombinant plasmids that contain only the insulin gene. Molecular biologists call this approach shotgun cloning, because many fragments are randomly cloned at once and no individual gene is specifically targeted for cloning. How would you know which recombinant plasmid contained the insulin gene? Moreover, if the insulin gene, or adjacent sequences, does not have restriction sites for the restriction enzyme you used, you might not have any recombinant plasmids containing the insulin gene. Even if you did create plasmids with the insulin gene, how would you separate these from the other recombinant plasmids? So how do you find a particular gene of interest and clone only the DNA sequence that you want to study? These questions can often be answered by a cloning approach involving DNA libraries. Creating DNA libraries, building a collection of cloned genes. Many cloning strategies begin by preparing a DNA library, a collection of cloned DNA fragments from a particular organism contained within bacteria or viruses as the hosts. Libraries can be saved for relatively long periods of time and screened to pick out different genes of interest. Two types of libraries are typically used for cloning: genomic DNA libraries and complementary DNA libraries, cDNA libraries. Figure 3.4 on the next page shows how genomic libraries and cDNA libraries are constructed. Genomic versus cDNA libraries. In a genomic library, chromosomal DNA from the tissue of interest is isolated and then digested with a restriction enzyme, see Figure 3.4a. This process produces fragments of DNA that include the organism's entire genome. A plasmid, BAC, YAC, or bacteriophage vector is digested with the same enzyme, and DNA ligase is used to ligate genomic DNA pieces and vector DNA randomly. In theory, all DNA fragments in the genome will be cloned into a vector. Recombinant vectors are then used to transform bacteria, and each bacterial cell clone will contain recombinant vector with a plasmid containing a genomic DNA fragment. Consider each clone a book in this library of DNA fragments. One disadvantage of creating this type of library for eukaryotic genes is that non-protein coding pieces of DNA, called introns, are cloned in addition to protein coding sequences, exons. Because a majority of DNA in any eukaryotic organism consists of introns, many of the clones in a genomic library will contain non-protein coding pieces of DNA. Another limitation of genomic libraries is that many organisms, including humans, have such large genomes that searching for a gene of interest would be like searching for a needle in a haystack. In a cDNA library, mRNA from the tissue of interest is isolated and used for making the library. However, mRNA cannot be cut directly with restriction enzymes, so it must be converted to a double-stranded DNA molecule. An enzyme called reverse transcriptase, RT, is used to catalyze the synthesis of single-stranded DNA from the mRNA, see Figure 3.4b. This enzyme is made by viruses called retroviruses, so named because they are exceptions to the usual flow of genetic information. Instead of having a DNA genome that can be used to make RNA, retroviruses have an RNA genome. After infecting host cells, they use RT to convert RNA into DNA, so that they can replicate. Human immunodeficiency virus, HIV, the causative agent of acquired immunodeficiency syndrome, AIDS, is a retrovirus. As we will discuss, retroviruses also have important applications in biotechnology as gene therapy vectors, Chapter 11. Because RT synthesizes DNA that is an exact copy of mRNA, it is called complementary DNA, cDNA. The mRNA is degraded by treatment with an alkaline. Figure 3.4 Comparison of a human genomic DNA library and a cDNA library. A. Human DNA is cleaved with a restriction enzyme to create a series of smaller fragments that are cloned into plasmids or other vectors. A. Human genomic library consists of a collection of bacteria each containing a different fragment of human DNA. In theory, all DNA fragments from the genome will be represented in the library. B. In a cDNA library, mRNA is converted into cDNA by the enzyme reverse transcriptase. Linkers containing a restriction site are added to the cDNA to create cohesive ends. The cDNA can now be cloned into a plasmid for subsequent replication in bacteria. Solution or enzymatically digested, then DNA polymerase is used to synthesize a second strand to create double-stranded cDNA. Because cDNA sequences do not necessarily have a convenient restriction site at each end, short, double-stranded DNA sequences called linker sequences are often enzymatically added to the ends of the cDNA. Linkers contain restriction sites. Different linkers for different restriction sites are commercially available. By adding linkers, cDNA can now be ligated into a convenient restriction site in a vector of choice, often a plasmid. The recombinant plasmid is then used to transform bacteria. One primary advantage of cDNA libraries over genomic libraries is that they are a collection of actively expressed genes in the cells or tissue from which the mRNA was isolated. Also, introns are not cloned in a cDNA library. By contrast, when cloned genomic DNA containing introns and exons is inserted into bacteria, the cells cannot splice mRNA transcribed from this DNA and remove introns. For this reason, cDNA libraries are typically preferred over genomic libraries when attempting to clone and express a gene of interest. Another advantage of cDNA libraries is that they can be created and screened to isolate genes that are primarily expressed only under certain conditions in a tissue. For example, if a gene is expressed only in a tissue stimulated by a hormone, researchers make libraries from hormone-stimulated cells to increase the likelihood of cloning hormone-sensitive genes. Libraries have become such a routine aspect of molecular biology that many companies sell libraries prepared from a range of tissues from different species. One disadvantage is that cDNA libraries can be difficult to create and screen if a source tissue with an abundant amount of mRNA for the gene is not available. But as you will learn, a technique called the polymerase chain reaction, PCR, can frequently solve this problem. Library screening. Once either a genomic library or a cDNA library is created, it must be screened to identify the genes of interest. One of the most common library screening techniques is called colony hybridization, figure 3.5. In colony hybridization, bacterial colonies from the library containing recombinant DNA are grown on an agar plate. A nylon or nitrocellulose membrane is placed over the plate, and some of the bacterial cells attach to the membrane at the same location where they are found on the plate. If bacteriophage vectors are used, phages are transferred onto the nylon. The nylon is treated with an alkaline solution to lyse bacteria and denature their DNA, which binds to the nylon as single-stranded molecules. Typically, the nylon is then incubated with a DNA probe, a single-stranded DNA fragment that is complementary to the gene of interest because it can base pair by hydrogen bonding to the target DNA to be cloned. Probes are tagged or labeled using either a radioactive nucleotide or, more commonly, a fluorescent dye or other compounds that can be used to catalyze light-releasing reactions called chemiluminescence. The dye makes it possible to track the probe to determine where it binds. The probe binds to complementary sequences on the nylon. A process is called hybridization. Figure 3.5 Colony hybridization, library screening with a DNA probe to identify a cloned gene of interest. The nylon is then washed to remove excess unbound probe and exposed to photographic film in a process called autoradiography. Anywhere the probe has bound to the filter, radioactivity from radioactive probes or released light, fluorescence or chemiluminescence, from non-radioactive probes exposes silver grains in the film. Depending on the abundance of the gene of interest, there may be only a few colonies, or plagues, on the filter that hybridize to the probe. Film is developed to create a permanent record, called an autoradiogram, or autoradiograph, which is then compared with the original plate of bacterial colonies to identify which colonies contained recombinant plasmid with the gene of interest. These colonies can now be grown on a larger scale to isolate the cloned DNA. Often, when hybridization is done using fluorescent or chemiluminescent probes, a digital imaging instrument can be used to detect probe binding and then a photograph aligned with the bacterial plate to identify colonies of interest. The type of probe used for library screening often depends on what is already known about the gene of interest. For example, the screening probe is frequently a gene cloned from another species. A cDNA clone of a gene from a rat or mouse is often a very effective probe for screening a human genomic or cDNA library, because many gene sequences in rats and mice are similar to those found in human genes. If the gene of interest has not been cloned in another species but some information is available about the protein sequence, a series of chemically synthesized oligonucleotides can be made based on a prediction of codons that can code for the known protein sequence. If some partial amino acid sequence is known for a protein encoded by a gene to be cloned, it is possible to work backward and design oligonucleotides based on the predicted nucleotides that coded for the amino acid sequence. In addition, if an antibody is available for the protein encoded by the gene of interest, an expression library, which results in protein expression in bacteria, can be used, and the library can be screened with the antibody to detect colonies expressing the recombinant protein. Library screening rarely results in the isolation of clones that contain full-length genes. It is more common to obtain clones with small pieces of the gene of interest. One reason why this occurs with cDNA libraries is because it may be difficult to isolate full-length mRNA or synthesize full-length cDNA for the gene of interest. When small pieces of a gene are cloned, scientists sequence these pieces and look for sequence overlaps. Overlapping fragments of DNA can then be pieced together like a puzzle in an attempt to reconstruct the full-length gene. This often requires screening the library several times, with large numbers of bacteria being plated and used for colony hybridization. Looking for start and stop codons in the sequenced pieces is one way to predict if the entire gene has been pieced together. Through this process, overlapping fragments can be pieced together to assemble an entire gene. Later in this chapter we will discuss how whole genome shotgun sequencing strategies can enable scientists to sequence entire genomes. Because of genomic studies, libraries are becoming a less common way to identify and clone genes. Instead of using a library to identify one or a few genes at a time, Genomics enables scientists to identify sequences for all genes in a genome. Polymerase chain reaction. Although libraries are very effective and commonly used for cloning and identifying a gene of interest, the polymerase chain reaction, PCR, is a much more rapid approach to cloning than building and screening a library. PCR is often the technique of choice when cloning genes, but it also has many other applications. Developed in the mid-1980s by Kary Mullis, PCR turned out to be a revolutionary technique that has had an impact on many areas of molecular biology. In 1993, Mullis won the Nobel Prize in Chemistry for his invention. PCR is a technique for making copies or amplifying a specific sequence of DNA in a short period of time. The concept behind a PCR reaction is remarkably simple. Target DNA to be amplified is added to a thin-walled tube and mixed with deoxyribonucleotides, DATP, DCTP, DGTP, DTTP, buffer, and DNA polymerase. A paired set of primers is added to the mixture. Primers are short single-stranded DNA oligonucleotides, usually around 20 to 30 nucleotides long. These primers are complementary to nucleotides flanking opposite ends of the target DNA to be amplified, figure 3.6. The reaction tube is then placed in a thermal cycler. In the simplest sense, a thermal cycler is a sophisticated heating block that is capable of rapidly changing temperature over very short time intervals. The thermal cycler takes the sample through a series of reactions called a PCR cycle, figure 3.6. Each cycle consists of 3 stages. In the first stage, called denaturation, the reaction tube is heated to approximately 94 degrees Celsius to 96 degrees Celsius, causing separation of the target DNA into single strands. In the second stage, called hybridization or annealing, the tube is then cooled slightly to between 55 degrees Celsius and 65 degrees Celsius, which allows the primers to hydrogen bond to complementary bases at opposite ends of the target sequence. During extension, or elongation, the last stage of a PCR cycle, the temperature is usually raised slightly, to about 70 degrees Celsius to 75 degrees Celsius, and DNA polymerase copies the target DNA by binding to the 3 ends of each primer and using the primers as templates. DNA polymerase adds nucleotides to the 3 end of each primer to synthesize a complementary strand. At the end of one complete cycle, the amount of target DNA has been doubled. The thermal cycler. Figure 3.6. The polymerase chain reaction repeats these 3 stages again according to the total number of cycles determined by the researcher, usually 20 or 30 cycles. The greatest advantage of PCR is its ability to amplify millions of copies of target DNA from a very small amount of starting material in a short period of time. Because the target DNA is doubled after every round of PCR, after 20 cycles of PCR, approximately 1 million copies, 220, of target DNA are produced from a reaction starting with 1 molecule of target DNA. One key to PCR is the type of DNA polymerase used in the reaction. The repeated heating and cooling required for PCR would denature and destroy most DNA polymerases after just a few cycles. Several sources of PCR-suitable DNA polymerases are available. One of the first and most popular enzymes for PCR is known as TAC DNA polymerase. TAC is isolated from the domain archaea called Thermus aquaticus, a species that thrives in hot springs. Because T. aquaticus is adapted to live in hot water, it was first discovered in the hot springs of Yellowstone National Park. It has evolved a DNA polymerase that can withstand high temperatures. Because TAC is stable at high temperatures, it can withstand the temperature changes necessary for PCR without being denatured. Thermostable polymerases such as TAC are essential for PCR. There are many variations and different applications in PCR technology. For example, real-time or quantitative PCR, qPCR, uses specialized thermal cyclers that enable researchers to quantify amplification reactions as they occur. We will discuss qPCR later in this chapter. An excellent tutorial on PCR can be viewed at the Cold Spring Harbor DNA Learning Center website listed at the companion website. PCR has widespread applications in research and medicine, such as making DNA probes, studying gene expression, amplifying minute amounts of DNA to detect viral pathogens and bacterial infections, amplifying DNA to diagnose genetic conditions, detecting trace amounts of DNA from tissues at a crime scene, and even amplifying ancient DNA from fossilized dinosaur tissue. Figure 3.7. Many PCR applications are described throughout the book. Cloning PCR products. PCR is often used instead of library screening approaches for cloning a gene because it is rapid and effective. Figure 3.8. A disadvantage of PCR cloning is that, to design primers, you need to know something about the DNA sequences that flank your gene of interest. Cloning by PCR is easiest if the gene has already been cloned in another species, for instance, using primers for a gene cloned previously from mice to clone the equivalent gene from humans. There are many ways to clone a gene using PCR. One modern approach to PCR cloning takes advantage of an interesting quirk of thermostable polymerases. As DNA is copied, TAC and other polymerases used for PCR normally add a single adenine nucleotide to the 3' end of all PCR products. Figure 3.8. After amplifying a Figure 3.7 PCR applications. The amplification of DNA by PCR has become an essential technique in molecular biology with a wide range of different applications. Examples of some of the more common biotechnology-related applications are represented in this figure. Figure 3.8 Cloning a gene by PCR. Target gene. Cloned PCR products can be ligated into plasmids called T vectors. T vectors contain a single-stranded thymine nucleotide at each end that can complementarily base pair with overhanging adenine nucleotides in PCR products. Once ligated into a T vector, the recombinant plasmid containing the cloned PCR product can be introduced into bacteria and its nucleotide sequence can be determined. Now that you have learned some of the most common strategies used to clone genes, in the next section we will consider a wide range of different approaches that scientists use to study cloned genes. 3.4 Laboratory techniques and applications of recombinant DNA technology. Why clone DNA? What can you do with a cloned gene? There are numerous applications of gene cloning and recombinant DNA technology. Figure 3.9 summarizes common gene cloning applications, many of which are discussed further in other chapters. In this section, we present some important routine molecular biology laboratory techniques and basic applications of gene cloning. Agarose gel electrophoresis. Agarose gel electrophoresis is one of the most common laboratory techniques used in working with DNA because it allows one to separate and visualize DNA fragments based on size. Figure 3.10 on the next page. Agarose is a material that is isolated from seaweed, melted in a buffer solution, and poured into a plastic tray. As the agarose cools, it solidifies to form a horizontal semi-solid gel containing small holes or pores through which DNA fragments will travel. The percentage of agarose used to create the gel determines its ability to resolve DNA fragments of different sizes. Most applications generally involve gels that contain 0.5% to 2% agarose. A gel with a high percentage of agarose, say 2%, is better suited for separating small DNA fragments because they will snake their way through the pores more easily than large fragments, which do not separate through the dense gel very well. A lower percentage of agarose is better suited for resolving large DNA fragments. To run a gel, it is submerged in a buffer solution that will conduct electricity. DNA samples are loaded into small depressions called wells, and an electric current is applied through electrodes at opposite ends of the gel. Separating DNA by electrophoresis is based on the fact that DNA migrates through a gel according to its charge and size, in base pairs. The sugar phosphate backbone renders DNA negatively charged, therefore, when DNA is placed in an electrical field, it migrates toward the anode, positive pole, and is repelled by the cathode, negative pole. Because all DNA is negatively charged regardless of the length or source, the rate of DNA migration and separation through an agarose gel depends on the size of a DNA molecule. Migration distance is inversely proportional to the size of a DNA fragment, so large DNA fragments migrate short distances. Figure 3.9 Applications of recombinant DNA technology. Figure 3.10 Agarose gel electrophoresis. A. DNA fragments can be separated and visualized by agarose gel electrophoresis. B. Photograph of an agarose gel stained with ethidium bromide. Lanes labeled as marker DNA were loaded with commercially prepared DNA size standards. These serve as a ladder of fragments of known size that are used to determine the size of experimental samples of DNA being analyzed. The lane labeled lambda DNA uncut shows high molecular weight uncut chromosomal DNA from phage lambda DNA plus hind III shows a series of discrete fragments created when lambda DNA is digested with the restriction enzyme hind III. The lane labeled E. coli DNA uncut contains undigested chromosomal DNA, and the adjacent lane shows E. coli chromosomal DNA digested with hind III, E. coli DNA and hind III. Through a gel relatively slowly and small fragments migrate faster. Tracking dyes are added to monitor DNA migration during electrophoresis. After the desired time of electrophoresis, DNA in the gel is stained using dyes such as ethidium bromide, which intercalate, penetrate, in between the base pairs of DNA. These dyes fluoresce when they are exposed to ultraviolet light. A permanent record of the gel is obtained by photographing the gel while it is exposed to ultraviolet light, figure 3.10b. Notice how the Hindith digested E. coli DNA produces a smear of bands unlike the set of discrete fragments visualized with Hindith digested lambda DNA. This smearing is due to the large size of the E. coli chromosome and the large number of cutting sites for hind III. So many fragments are created that it is not possible to visualize discrete bands. You will encounter techniques involving agarose gel electrophoresis throughout this book and, as a student, you are very likely to learn to run agarose gels during your biotechnology training. Restriction mapping gene structure. In the early days of gene cloning, soon after a gene was cloned, typically a type of physical map of the gene would be created to determine which restriction enzymes cut the cloned gene and to pinpoint the location of these cutting sites. Knowing the restriction map of a gene was very useful for making clones of small pieces of the gene, called subcloning, and manipulating many relatively small pieces of DNA, for example, 100 to 1000 BP, to sequence DNA and prepare DNA probes to study gene expression. To create a restriction map, cloned DNA is subjected to a series of single digests with restriction enzymes as well as double digests with combined enzymes. The digested DNA is then separated by agarose gel electrophoresis. Once the DNA samples have been digested, separated, and visualized by gel electrophoresis, creating the actual restriction map is like assembling a puzzle. As illustrated in figure 3.11, by comparing the single digests with each double digest, researchers can arrange the fragments in the correct order to create a map of restriction sites. Because DNA sequencing of even relatively small pieces of DNA has become fairly common, restriction mapping is now typically done using bioinformatics software, such as WebCutter, described in Problem 8 in the questions and activities at the end of this chapter, to identify restriction cutting sites in a DNA sequence. Figure 3.11 Restriction Mapping. The location of restriction sites for BAM-HI and PSTI is determined in a DNA fragment that is 7 kb long. Digestion with BAM-HI cleaves the DNA into two fragments measuring 2.5 and 4.5 kb, indicating that the DNA was cut at a single site located 2.5 kb from one end. Digestion with PSTI cleaves the DNA into two fragments at 1.5 and 5.5 kb, indicating that the DNA was cut at a single site located 1.5 kb from one end. A double digest with both enzymes cleaves the DNA into three fragments, 3.0 kb, 2.5 kb, and 1.5 kb. Because the 3.0 kb fragment is not created by either BAM-HI or PSTI digestion alone, it must represent the DNA located between the BAM-HI and PSTI cutting sites. Once this puzzle of fragments has been arranged, it becomes clear that the restriction map at the bottom of the figure is the only map consistent with the pattern of fragments created by the digests in this experiment. Without having to actually digest DNA and create a map experimentally. However, because of the historical importance of restriction mapping and its occasional use today, it is still of value to be aware of this technique. DNA sequencing. After a gene is cloned, it is important to determine the nucleotide sequence of the gene, its exact order of S, GS, TS, and CS. Knowing the DNA sequence of a gene can be helpful. 1. To deduce the amino acid sequence of a protein encoded by a cloned gene. 2. To determine the exact structure of gene. 3. To identify regulatory elements such as promoter sequences. 4. To identify differences in genes created by gene splicing. And 5. To identify genetic mutations, among other reasons. Different methods of DNA sequencing are available, including techniques for PCR cycle sequencing and computer-automated DNA sequencing, and these technologies are rapidly improving each year. Initially, the most widely used sequencing approach was chain termination sequencing, a manual method developed in 1977 by Frederick Sanger and often referred to as the Sanger method. In this technique, a DNA primer was hybridized to denatured template DNA, such as a recombinant plasmid, to be sequenced in a reaction tube containing deoxyribonucleotides and DNA polymerase. Because many modern plasmids are designed with sequencing primer binding sites adjacent to the multiple cloning site, DNA polymerase can be used to extend a complementary strand from the 3' end of primers hybridized to the plasmid. The original approach utilized radioactively labeled primer sequences. A small amount of a modified nucleotide called a dideoxyribonucleotide, DDNTP, was mixed in with the vector, primer, polymerase, and deoxyribonucleotides. A DDNTP differs from a normal deoxyribonucleotide, DNTP, because it has a hydrogen group attached to the 3-carbon of the deoxyribose sugar instead of a hydroxyl group O. See figure 3.12 on the next page. When a DDNTP is incorporated into a chain of DNA, the chain cannot be extended because the absence of a 3O prevents the formation of a phosphodiester bond with a new nucleotide, hence, the chain is terminated. In the original Sanger approach, 4 separate reaction tubes were set up. Each tube contained vector, primer, and all 4 DNTPs, but each tube also contained a small amount of 1-DDNTP. As synthesis of a new DNA strand from the primer begins, DNA polymerase randomly inserts a DDNTP into the sequence instead of a normal DNTP, preventing further synthesis of a complementary strand. Over time, a DDNTP will be incorporated at all positions in the newly synthesized strands, creating a series of fragments of varying lengths that are terminated at dideoxy residues. For the original Sanger technique, the DNA strands were separated on a thin polyacrylamide gel, which can separate sequences that differ in length by a single nucleotide. Autoradiography was used to detect the radioactive sequencing fragments shown in figure 3.12c. The sequence determined from the autoradiogram is read from the bottom to the top as individual nucleotides. As shown in figure 3.12c, the sequence determined from the autoradiogram is complementary to the sequence on the template strand in the vector. Figure 3.12 Computer Automated DNA Sequencing. A. The structure of a dideoxynucleotide, ddNTP. Note that the 3 group attached to the carbon is a hydrogen rather than a hydroxyl group, O. Because another nucleotide cannot be attached at the 3 end of a ddNTP, these nucleotides are the key to DNA sequencing by the Sanger method as shown. B. Computer-automated approaches separate DNA fragments using a capillary gel. A laser and detector are used to detect fluorescence of each dideoxynucleotide. C. An autoradiogram from an original dideoxy sequencing reaction in which radioactive ddNTPs or primers were used and 4 separate reactions carried out. The letters over the lanes, A, C, G, and T, correspond to the particular ddNTP used in the sequencing reaction analyzed in the lane. Analyzing Sanger sequencing by autoradiography is largely obsolete, having been replaced by computer-automated sequencing approaches as shown in B, and next-generation sequencing technologies. You decide to patent or not. The Human Genome Project was completed ahead of schedule in part because of competition between publicly funded genome centers and privately funded companies such as Celera Genomics, originally directed by former NIH researcher Craig Venter. While at the Institute for Genomic Research, TIGR, Venter and colleagues were the first scientists to completely sequence the genome of a living organism, the bacterium Haemophilus influenza. This group applied for patents on the nucleotide sequence for H. influenza and on the bioinformatics technology used to analyze this genome. Previously, Venter and his colleagues described a set of experiments in which they randomly cloned short pieces of CNS from human brain cells. These short sequences, called expressed sequence tags, ESTs, could in theory be used as probes to identify full-length CNS. Some of Venter's ESTs were found to be identical to genes that had already been cloned or to a portion of a gene, others appeared to be novel gene sequences or junk DNA. Hoping to gain proprietary rights to full-length genes that could be identified from Venter's ESTs, TIGR applied for a patent. This request generated a great deal of controversy. For instance, should scientists be allowed to patent DNA sequences from naturally living organisms? What if a patent is awarded for only small pieces of a gene even if no one knows what a DNA sequence does just because some individuals or a company wants a patent to stake their claim to having cloned a piece of DNA first? What if there are no clear uses for the DNA sequences cloned? Can or should investigators who use a gene microarray or create a DNA library be allowed to patent the entire genome of any organism they have studied? When they are granted a patent, scientists essentially have a monopoly on patented information for 2 decades from the patent filing date. Many believe that the hoarding of genome information is against the tradition of sharing information to advance science. Would awarding patents slow progress in cloning genes if groups hoarded data and did not share information? Could or should a group stake a claim to a gene, thereby preventing others from working on it or developing a product from it? What about individuals who figure out what to do with the gene? Should a genetically engineered living organism be patented? Engineered bacteria, for example, those used to clean up environmental pollution, and transgenic animals have been patented, as have clinically important genes such as beta-interferon. Can a group claim rights to anticipated future uses of the gene even if there are no data to substantiate such claims? What if a gene sequence is involved in a disease for which a genetic therapy may be developed? Since 1980, the U.S. Patent and Trademark Office has granted patents on more than 35,000 genes or gene sequences, and an estimated 20% of human genes have been patented. Some scientists are concerned that patents awarded for simply cloning a piece of DNA is awarding a patent for too little work. Many scientists believe it is more appropriate to patent the novel technology used to discover and study genes and the applications of genetic technology such as gene therapy approaches than to patent the gene sequences themselves. Generally, most gene patents cover technology uses for a sequence, such as a genetic test, and not the DNA sequence itself. From a commercial standpoint, one advantage of patenting is that it provides private companies with a financial incentive to get a medicine or technology to the marketplace and enables them to make a profit after many millions, or billion in some cases, of dollars are spent on R&D to make a product. To patent or not? You decide. High-throughput computer automated sequencing replaces the original Sanger sequencing technique. Because of limitations in running a sequencing gel, the original Sanger method could be used only to sequence approximately 200 to 400 nucleotides in a single reaction. Therefore, in sequencing a longer piece of DNA, for example, 1,000 base pairs, it was necessary to run multiple reactions to create overlapping sequences that could be pieced together to determine the entire, continuous sequence of 1,000 base pairs. Because of this limitation, the Sanger sequencing approach was a cumbersome method for the large-scale sequencing efforts such as those used for the Human Genome Project. The project was completed ahead of schedule in part because of the development of computer-automated DNA sequencing methods capable of sequencing long stretches of DNA, more than 500 BP in a single reaction. Initially, computer-automated sequencing reactions used either DD and TPS, each labeled with a different colored non-radioactive fluorescent dye, or a sequencing primer labeled at its 5 end with a dye, figure 3.12. A single reaction tube is used, and the manual-style approach of using polyacrylamide gels followed by autoradiography has been replaced by separating sequencing reactions on a single lane of an ultra-thin diameter tube gel called a capillary gel. As DNA fragments move through the gel, they are scanned with a laser beam. The laser stimulates the fluorescent dye on each DNA fragment, which emits different wavelengths of light for each DD and TP. The emitted light is collected by a detector that amplifies and then feeds this information to a computer to process and convert the light patterns and reveal the DNA sequence, figure 3.13. Automated DNA sequencers often contain multiple capillary gels several feet long, allowing many bases to be separated. Therefore, some instruments run as many as 96 capillary gels, each producing around 900 bases of sequence. With these instruments, it became possible to generate approximately 2 million bases of sequence in a day. As a result, these sequencers became known as high-throughput sequencers because of their ability to process and generate large amounts of sequence data in relatively short periods of time. Next-generation sequencing, NGS. Genomics has spurred a demand for sequencers that are faster and capable of generating millions of bases of DNA sequence in a relatively short time, leading to the development of next-generation sequencing, NGS, technologies designed to produce highly accurate and long stretches of DNA sequence, greater than 1 gigabase, billion bases, of DNA per reaction, at a low cost. Next-generation sequencing approaches dispense with the Sanger technique and capillary electrophoresis methods in favor of sophisticated, parallel formats, simultaneous reaction formats, that use state-of-the-art fluorescence imaging techniques. NGS technologies are providing an unprecedented capacity for generating massive amounts of DNA sequence data rapidly, up to 200 times faster than Sanger approaches in some cases, and at dramatically reduced costs per base. The desire for next-generation sequencing among the research community and challenges such as the $1,000 genome, discussed later in this chapter, has led to an intense technology race among many companies eager to produce NGS methods. In 2005, Roche 454 Life Systems was the first company to commercialize a next-generation sequencing technology, figure 3.13. This approach sequences genomes using a so-called solid-phase method in which beads are attached to fragmented genomic DNA, which is then PCR amplified in separate water droplets in oil for each bead, loaded into multi-well plates, and mixed with DNA polymerase. Multi-well plates often contain more than 1 million wells with 1 bead per well, each serving as a reaction tube for sequencing, see figure 3.13. A reaction called pyrosequencing is then used to sequence DNA on the beads in each well. In pyrosequencing, a single labeled nucleotide, e.g., DGTP, is flowed over the wells. Each well contains a single bead along with primers annealed to the DNA on the bead. When a complementary nucleotide crosses a template strand adjacent to the primer, it is added to the 3' end of the primer by DNA polymerase. Incorporation of a nucleotide results in the release of pyrophosphate, which initiates a series of chemiluminescent, light-releasing, reactions that ultimately produce light by using the firefly enzyme luciferase. Emitted light from the reaction is captured and recorded to determine when a single nucleotide has been incorporated into a strand. By rapidly repeating the nucleotide flow step with each of the 4 nucleotides to determine which base is next in the sequence, this approach can generate read lengths of about 400 bases and on the order of 400 million bases, MB, of data per 10-hour run. NGS sequencing technologies are also creating major data management challenges for saving and storing such large image data files. The company Applied Biosystems, ABI, has developed an approach called SOLID, supported oligonucleotide ligation and detection, that can produce 6 gigabases of sequence data per run. The SOLID method combines a variety of approaches to sequence DNA fragments that are linked to beads and amplified, similar to the 454 approach, but different sequencing technologies are used, which can provide greater output of sequencing data per instrument run. The instrumentation to run these platforms is expensive, but given the massive amounts of sequence that the NGS methods can generate, the average cost per base is much lower than that of Sanger sequencing. Through 2006, new sequencing technologies were cutting sequencing costs in half about every 2 years. Work on third-generation sequencers is actively underway, and there is every reason to believe that these instruments will be available by the middle part of this decade. For example, one promising approach on the immediate horizon involves the use of nanotechnology by pushing single-stranded DNA. Figure 3.13 Roche 454 Next-Generation Sequencing Technology. Roche 454 sequencing technology binds DNA fragments to beads. DNA fragments are amplified by PCR and then added to wells, step 1, and subjected to pyrosequencing, as described in the text. Step 2, during pyrosequencing, an individual fluorescent nucleotide, the blue G shown in this case, is flowed over the well. As a nucleotide is added to a primer by DNA polymerase, inorganic pyrophosphate, PPI, is released, which reacts with adenosine 5-phosphosulfate, APS, and the enzyme sulforylase to produce ADP, blue arrow. The firefly enzyme luciferase uses ADP to generate light, which can be detected and quantified. Step 3. Light captured by a detection system is analyzed to trace the pattern of nucleotides added to each well. The flow cycle is subsequently repeated with each of the other 3 nucleotides. Continual cycling of this process generates sequencing reads of approximately 400 bases. Fragments into nanopores and then cleaving off individual bases to produce a signal that can be captured. This method does not involve DNA amplification or fluorescent tags and thus provides direct sequencing of the DNA in a single strand. Fluorescence in situ hybridization. A technique called fluorescence in situ hybridization, FISH, in situ means in place, can be used to identify which chromosome contains a gene of interest. For example, if you just cloned a human gene believed to be involved in intelligence, you could use FISH to determine on which chromosome this gene resides. In FISH, chromosomes are isolated from cells such as white blood cells and spread out on a glass microscope slide. A DNA or RNA probe for the gene of interest is labeled with fluorescent nucleotides and then incubated in solution with the slide. The probe hybridizes with complementary sequences on chromosomes on the slide. The slide is washed and then exposed to fluorescent light. Wherever the probe has bound to a chromosome, the fluorescently labeled probe is illuminated to indicate the presence of probe binding. Figure 3.14. To determine which of the 23 human chromosomes show fluorescence, they are aligned according to the length and staining patterns of their chromatids to create a karyotype. Fluorescence on more than one chromosome indicates either multiple copies of the gene or related sequences that may be part of a gene family. FISH is also used to analyze genetic disorders. For example, FISH analysis can be performed on a karyotype of fetal chromosomes from a pregnant woman to determine if a developing fetus has an abnormal number of chromosomes. When a gene is expressed in an organ with many different cell types, for example, kidney or brain tissue, FISH can also be used to determine the cell type that is expressing a particular mRNA. In this approach, the tissue of interest is preserved in a fixative solution and then embedded in a wax-like material or resin. This allows researchers to slice the tissue into thin sections about 1 to 5 mm thick and to attach them to a microscope slide. Sometimes frozen sections of tissue are used for these experiments. The slide is incubated with a fluorescent dye-tagged RNA or DNA probe for the gene of interest. The probe hybridizes to mRNA within cells in their native place, and probe binding is determined by detecting fluorescence. For some studies, PCR can even be performed directly on tissue sections in situ as a way of determining cell type expression for a given gene. Figure 3.14 Fluorescence in situ hybridization. White spots at the tips of each chromosome indicate fluorescence from a probe binding to telomeres. Southern blotting. Another technique, called southern blot analysis, southern blotting or hybridization, is frequently used to determine gene copy number among other applications. Developed by Ed Southern in 1975, southern blotting begins by digesting chromosomal DNA into small fragments with restriction enzymes. DNA fragments are separated by agarose gel electrophoresis. Figure 3.15 Southern blot analysis of DNA fragments. A. Region of DNA for a gene of interest to be studied by southern blot analysis. B. Steps involved in southern blotting. Figure 3.15. However, the number of restriction fragments generated by digesting chromosomal DNA is often so great that simply running a gel and staining the DNA does not resolve discrete fragments. Rather, digested DNA appears as a continuous smear of fragments in the gel. Southern blotting is used to visualize only specific fragments of interest. Following electrophoresis, the gel is treated with an alkaline solution to denature the DNA, then the fragments are transferred onto a nylon or nitrocellulose membrane using a technique called blotting. Blotting can be achieved by setting up a gel sandwich in which the gel is placed under the nylon membrane, filter paper, paper towels, and a weight to allow for wicking of a salt solution through the gel, which will transfer DNA onto the nylon by capillary action. Figure 3.15. The single strand of DNA stick to the blot, positioned in bends exactly as on the gel. Alternatively, pressure or vacuum blotters can be used to transfer DNA onto nylon. The nylon blot is then baked or briefly exposed to UV light to attach the DNA permanently. Next, the blot is incubated with a labeled probe in much the same way that colony hybridizations are carried out. Increasingly, non-radioactive probes are being used for southern blotting and many other hybridization techniques. The blot is washed to remove extraneous probes and then exposed to film by autoradiography if a radioactive or chemiluminescent probe is used. Wherever the probe has bound to the blot, radioactivity or light released by the probe develops silver grains on the film to expose bands on the blot, creating an autoradiogram. See Figure 3.15. Chemiluminescent probes can also be detected by digital camera imaging systems. By interpreting the number of bands revealed, it can be possible to determine gene copy number. The development of southern blot analysis was an important technique, which formed the basic principles for northern blotting, the separation and blotting of RNA molecules, as discussed in the next section, and western blotting, the separation and blotting of proteins. Northern and western blotting techniques were not named after scientists named Northern and Western, rather, they were named as tongue-in-cheek references to Ed Southern, the founder of southern blots. Southern blotting has many other applications, including gene mapping, related gene family identification, genetic mutation detection, PCR product confirmation, and DNA fingerprinting. a topic we discuss in Chapter 8. An excellent animation of how southern blot analysis is used in DNA fingerprinting can be found at the website for the Cold Spring Harbor DNA Learning Center, which can be found on the companion website. Studying gene expression. Molecular biologists around the world are involved in research studying gene expression and the regulation of gene expression. Numerous different molecular techniques are available for studying gene expression. Most methods involve analyzing mRNA produced by a tissue. This is often a good measure of gene expression because the amount of mRNA produced by a tissue is often equivalent to the amount of protein the tissue makes. Northern blot analysis. The basic methodology of a northern blot is similar to southern blot analysis. In northern blotting, RNA is isolated from a tissue of interest and separated by gel electrophoresis. The RNA is not digested with enzymes. RNA is blotted onto a nylon membrane and then hybridized to a probe, as described for southern blots. Exposed bands on the autoradiogram show the presence of mRNA for the gene of interest and the size of the mRNA, figure 3.16a. In addition, the amounts of mRNA produced by different tissues can be compared and quantified. Reverse transcription PCR. Sometimes the amount of RNA produced by a tissue is below the level of detection by northern blot analysis. PCR allows for detecting minute amounts of mRNA from even very small amounts of starting tissue. For instance, PCR has been a great tool for molecular biologists studying gene expression in embryos and developing tissues where the amount of tissue for analysis is very small. Because RNA cannot be directly amplified by PCR, a technique called reverse transcription PCR, RT-PCR, is carried out. In RT-PCR, isolated mRNA is converted into double-stranded cDNA by the enzyme reverse transcriptase in a process similar to the way in which cDNA for a library is made. The cDNA is then amplified with a set of primers specific for the gene of interest. Amplified DNA fragments are electrophoresed on an agarose gel and evaluated to determine expression patterns in a tissue, see figure 3.16b. The amount of cDNA produced in a RT-PCR reaction for a particular gene of interest reflects the amount of mRNA, and thus the level of gene expression, for that particular gene in a given tissue. Real-time PCR. Real-time or quantitative PCR, qPCR, enables researchers to quantify amplification reactions as they occur in real time, figure 3.17 on page 86. There are several ways to run real-time PCR reactions, but the basic procedure involves the use of specialized thermal cyclers that use a laser to scan a beam of light through the top or bottom of each PCR tube. Each reaction tube contains either a dye-containing probe or DNA-binding dye that emits fluorescent light when illuminated by the laser. The light emitted by these dyes correlates with the amount of PCR product amplified. Light from each tube is captured by a detector, which relays information to a computer to provide a readout on the amount of fluorescence after each cycle. This readout can be plotted and analyzed to quantitate the number of PCR products produced after each cycle. Two commonly used approaches for real-time PCR involve the use of TPMAN probes and a dye called SYBR green. SYBR green binds double-stranded DNA. As more double-stranded DNA is copied with each round of real-time PCR, there are more DNA copies to bind SYBR green, which increases the amount of fluorescent light emitted. TPMAN probes are complementary to specific regions of the target DNA between where the forward and reverse primers for PCR bind, figure 3.17. TPMAN probes contain two dyes. One dye, the reporter, is located at the 5' end of the probe and can release fluorescent light when excited by laser light from the thermal cycler. The other dye, called a quencher, is figure 3.16 analyzing gene expression by northern blot analysis and RT-PCR. Northern blot analysis and RT-PCR are two common techniques for analyzing the amount of mRNA produced by a tissue, gene expression. Blot 1 is a portion of an autoradiogram from a northern blotting experiment in which RNA from 4 rat tissues 1, seminal vesicles, 2, kidney, 3 and 4, different segments of the epididymis, a male reproductive organ, was blotted onto nylon and then probed with a radioactive cDNA probe for a gene involved in protecting tissues from damage by harmful free radicals, atoms, or molecules with unpaired numbers of electrons. Notice how the amount of mRNA detected in lanes 3 and 4, as indicated by the size and darkness of each band, is greater than the amount of mRNA in lanes 1 and 2. Blot 2 shows an autoradiogram from blot 1 that was stripped of bound probe and reprobed with a probe for a different gene. Blot 3, the same blot shown in the other two panels, was stripped of bound probe and reprobed with a radioactive cDNA for a gene, cyclophilin, that is expressed at nearly the same levels in virtually all tissues. B. Agarose gel from an RT-PCR experiment in which RNA from rat tissues was reverse transcribed and amplified with primers for beta-actin, an important component of the cytoplasm of cells, and /or beta-defensin 1, a gene that encodes a peptide that provides protection against bacterial infections in many tissues. Lane 1 contains DNA size standards of known size, often called a ladder, increasing in 100 BP increments. Lane 2 is a negative control sample in which primers were added to a PCR experiment without cDNA. Lane 3 is a negative control sample in which cDNA was added to a PCR experiment without primers. Notice that lanes 2 and 3 do not show any amplified PCR product because amplification will not occur without cDNA as target DNA, lane 2, or without primers, lane 3. Lane 4 shows kidney cDNA amplified with actin primers. Lane 5 shows kidney cDNA amplified with defensin primers. Lane 6, 7, and 8 show PCR products from cDNA of 3 different rat reproductive tissues that were amplified with both actin and defensin primers. Notice how lanes 6 and 8 show relatively even amounts of actin and defensin PCR products. Greater amounts of defensin PCR product are shown in lane 7. These differences reflect the different amounts of defensin mRNA made by these tissues. Attached to the 3 end of the probe. When these two dyes are close to each other, the quencher dye interferes with the fluorescent light released from the reporter dye. However, as DNA polymerase extends each primer, it removes the reporter dye from the end of the probe, and eventually removes the entire probe. Now that the reporter dye is separated from the quencher, the fluorescent light released by the reporter can be detected by the thermal cycler. Detected light is analyzed by a computer to produce a plot displaying the amount of fluorescence emitted with each cycle. Because real-time PCR does not involve running gels, it is a powerful and rapid technique for measuring and quantitating changes in gene expression, particularly when multiple samples and different genes are being analyzed simultaneously. Gene microarrays. DNA microarray analysis is another technique for studying gene expression that rapidly gained popularity over the past 15 years because it enables researchers to analyze all the genes expressed in a tissue very quickly. Figure 3.18 on page 87. A microarray, also known as a gene chip, is created with the use of a small glass microscope slide. Single-stranded DNA molecules are attached or spotted onto the slide using a computer-controlled high-speed robotic arm called an arrayer, which is fitted with a number of tiny pins. Each pin is immersed in a small amount of solution containing millions of copies of different DNA molecules, such as CNS for different genes. The arrayer fixes this DNA onto the slide at specific locations, points or spots, recorded by a computer. A single microarray can have Figure 3.17 Real-time PCR. A. The Tickman method of real-time PCR involves a pair of PCR primers along with a probe sequence complementary to the target gene. The probe contains a reporter dye, R, at one end and a quencher dye, Q, at the other. When the quencher dye is close to the reporter dye, it interferes with fluorescence released by the reporter dye. When DNA polymerase extends a primer to synthesize a strand of DNA, it cleaves the reporter dye off the probe, allowing the reporter to give off energy. B. Each subsequent PCR cycle removes more reporter dyes, so that increased light emitted from the dye can be captured by a computer to produce a readout of fluorescence intensity with each cycle. Over 10,000 spots of DNA, each containing unique sequences of DNA for a different gene. There are several different kinds of microarrays. Figure 3.18 shows a representative example of how a microarray can be used to study gene expression. Scientists begin by extracting mRNA from a tissue of interest. Then cDNA synthesized from this mRNA is labeled with a fluorescent dye. Labeled cDNA is incubated overnight with the microarray where it hybridizes to spots on the microarray that contain complementary DNA sequences. The microarray is washed and scanned by a laser that causes the cDNA hybridized to the microarray to fluoresce. Fluorescent spots reveal which genes are expressed in the tissue of interest, and the intensity of fluorescence indicates the relative amount of expression. The brighter the spot, the more mRNA is expressed in that tissue. Microarrays can also be run by labeling thickness from two or more tissues with different colored fluorescent dyes. Gene expression patterns from the different tissues are compared based on the color of spots that appear following hybridization and detection. For many species, including humans, entire genomes are available on microarrays. Researchers are also using microarrays to compare patterns of expressed genes in tissues under different conditions. For example, cancer cells can be compared with normal cells to look for genes that may be involved in cancer formation. Results of such microarray studies can be used to develop new drug therapy strategies to combat cancer and other diseases. Chapter 11. Protein expression and purification. Bacteria that have been transformed with recombinant plasmids can often be used to produce the protein product of the isolated gene. Large quantities of bacteria can be grown in a fermenter, and the protein can be isolated. Chapter 4. Gene mutagenesis studies. Of the many different ways that scientists work with and study clone genes, there are a variety of techniques that can be used to study the structure and function of protein produced by a specific gene. One approach is called site-directed mutagenesis. In this technique, mutations can be created in specific nucleotides of a clone gene contained in a vector. The gene can then be expressed in cells, which results in the translation of a mutated protein. This allows researchers to study the effects of particular mutations on protein structure and serves as a way of determining which nucleotides are important for specific functions of the protein. Site-directed mutagenesis can be a very valuable way to help scientists identify critical sequences in genes that produce proteins involved in human diseases. RNA interference. In 1998, researchers Craig C. Mello of the University of Massachusetts Medical School and Andrew Z. Fire of Stanford University published groundbreaking work. Figure 3.18 Gene Microarray Analysis, in which they used double-stranded pieces of RNA, dsRNA, to inhibit or silence expression of genes in the nematode roundworm Sinorhabditis elegans. This naturally occurring mechanism for inhibiting gene expression is known as RNA interference, RNAi. As Mello, Fire, and other researchers discovered, dsRNA can be bound by an RNA-digesting enzyme called dicer, which cuts dsRNA into 21 to 25 nucleotide-long snippets of RNA molecules called small interfering RNAs, siRNAs. Figure 3.19 RNA interference. 1. Double-stranded RNA is cleaved by the enzyme dicer into siRNAs. 2. RISC proteins bind siRNAs and degrade one of the two strands. 3. To produce single-stranded siRNAs. 4. Single-stranded siRNAs bind to complementary sequences on mRNA molecules in the cytoplasm and interfere with, silence, gene expression through triggering mRNA degradation by slicer or by inhibiting translation of the mRNA by ribosomes. These siRNAs are bound by a protein-RNA complex called the RNA-induced silencing complex, RISC. RISC unwinds the double-stranded siRNAs, releasing single-stranded siRNAs that bind to complementary sequences in mRNA molecules. Binding of siRNAs to mRNA results in degradation of the mRNA, by the enzyme slicer, or blocks translation by interfering with ribosome binding. Figure 3.19. RNAi is similar in mechanism to the gene silencing actions of mRNAs, but a primary difference is that siRNAs are generated from dsRNA. See Chapter 2. Initially it appeared that the discovery of siRNAs in mRNAs was perhaps a relatively esoteric finding. Predictions now indicate that mammalian cells express thousands of siRNAs or mRNAs, and these may in turn regulate hundreds of genes. Biotechnology and pharmaceutical companies are looking for ways in which siRNAs and mRNAs may be exploited for therapeutic purposes. Techniques incorporating RNAi have developed rapidly as methods for regulating gene expression and as a potential way to target and inactivate specific genes with high efficiency. We'll consider examples of how biotechnology and pharmaceutical companies are working on RNAi techniques for silencing genes involved in human diseases later. Chapter 11. RNAi has become such a rapidly developing technology that recent estimates indicate the use of RNAi reagents will grow from about $400 million in 2005 to over $900 million by 2012. The 2006 Nobel Prize in Physiology or Medicine was awarded to Andrew Fire and Craig Mello for their discovery of this natural method for switching genes off. Nobel Prizes are typically awarded decades after the honored work has been completed, so this unusually quick recognition is a clear indication of the value of RNAi as a powerful and promising research tool. 3.5 Genomics and Bioinformatics, Hot Disciplines of Biotechnology. Cloning individual genes using libraries and other techniques described in this chapter will continue to be used in specialized approaches in recombinant DNA technology. However, over the last 15 years or so, a number of advances in cloning and sequencing technologies have increasingly led to the use of strategies for cloning, sequencing, and analyzing entire genomes in exciting and rapidly developing science called genomics. You will learn about many applications of genomics as you study biotechnology. Here we provide an introduction to how scientists can study whole genomes. Whole genome shotgun sequencing. As powerful as traditional recombinant DNA techniques are, it became increasingly apparent that if scientists wanted to study complex biological processes that involve many genes such as most cancers, cloning one or even a few genes at a time is too slow, yielding only incremental information about genes. As a result, scientists started working on strategies for cloning and sequencing entire genes, a strategy commonly called whole genome shotgun sequencing. The analogy is that cloning individual genes using libraries is equivalent to using a rifle to hit a specific spot on a target, e.g., cloning a specific gene, whereas the pellets from a shotgun would randomly hit many spots on a target with little precision. Figure 3.20 Whole genome shotgun sequencing and two examples of bioinformatics applications. Shown is a simplified example of a piece of genomic DNA cut into smaller pieces, labeled 1, 2, 3, and 4, by EchoRI and BAM-HI. Regardless of how a piece of DNA is cloned, DNA sequence analysis is an essential part of learning more about the cloned DNA. Typically such analysis begins by searching the unknown sequence against a database of known sequences. In this example, sequence alignment reveals that the unknown sequence cloned is an exact match with a sequence of a known gene already in the database. Note, for simplicity, only one strand of sequenced DNA is being compared with the database. Another application of bioinformatics involves the use of computer programs to align DNA fragments, contigs, based on nucleotide sequence overlaps. The scheme shown here is similar to one approach used in the human genome project to assemble completed sequences of entire chromosomes. Although when contigs are compared, typically only relatively short sequences at the end of each contig are aligned to assemble an entire chromosome. In shotgun sequencing, the entire genome, introns and exons, is sequenced. The complete genome sequence is assembled with the help of software programs, and then individual genes are identified through bioinformatics. One common shotgun strategy for constructing the sequence of whole chromosomes involves using restriction enzymes to digest pieces of entire chromosomes. Figure 3.20. This process can produce thousands of overlapping fragments called contigs, contiguous sequences, which have to be sequenced. Each contig is sequenced, and then computer programs are used to align the fragments based on overlapping sequence pieces, figure 3.20b. This approach enables scientists to reconstruct the entire sequence of a whole chromosome and, as you can see in the figure, the use of computers to organize and compare the DNA sequences of these fragments is essential. This is bioinformatics in action, an interdisciplinary field that applies computer science and information technology to promote an understanding of biological processes. In 1995, scientists at the Institute for Genomic Research were the first to use a shotgun approach to successfully sequence the genome of any organism when they sequenced the 1.8 million base pair genome of the bacterium Haemophilus influenza. Many doubted that shotgun sequencing strategies could be used effectively to sequence larger genomes, but development of this technique and novel sequencing strategies rapidly accelerated the progress of the Human Genome Project. Bioinformatics, merging molecular biology with computing technology. When scientists sequence a newly identified gene or DNA sequence, they report their findings in scientific publications and submit the sequence data to databases so that other scientists who may be interested in this sequence information can have access to it. Database manipulations of DNA sequence data were among the first applications of bioinformatics, which involves the use of computer hardware and software to study, organize, share, and analyze data related to gene structure, gene sequence and expression, and protein structure and function. As genome data have rapidly accumulated, resulting in an enormous amount of information being stored in public and private databases, bioinformatics has become an essential tool that allows scientists to share and compare data, especially DNA sequence data. We discuss basic applications of bioinformatics throughout this book, however, we introduce the field here with a few very common applications. Examples of bioinformatics in action. Even before whole genome sequencing projects began, scientists were accumulating sequence information from a variety of different organisms, necessitating the development of sophisticated databases that could be used by researchers around the world to store, share, and obtain the maximum amount of information from protein and DNA sequences. Databases are essential tools for archiving and sharing data with researchers and the public. Because high-speed computer-automated DNA sequencing techniques were developed nearly simultaneously with expansion of the Internet as an information tool, many DNA sequence databases became available through the Internet. When scientists who have cloned a gene enter their sequence data into a database, these databases search the new sequence against all other sequences in the database and create an alignment of similar nucleotide sequences if a match is found. Figure 3.20a. This type of search is often one of the first steps taken after a gene is cloned using recombinant DNA techniques because it is important to determine if the sequence has already been cloned and studied or if the gene sequence is a novel one. Among other applications, these databases can also be used to predict the sequence of amino acids encoded by a nucleotide sequence and to provide information on the function of the cloned gene. One of the most widely used DNA sequence databases worldwide is called GenBank. It is the largest publicly available database of DNA sequences and contains the National Institutes of Health (NIH) collection of DNA sequences. GenBank shares and acquires data from databases in Japan and Europe. Maintained by the National Center for Biotechnology Information (NCBI) in Washington, D.C. GenBank contains more than 100 billion bases of sequence data from over 100,000 species, and it is growing rapidly every year. The NCBI is a goldmine of bioinformatics resources that creates public access databases and develops computing tools for analyzing and sharing genome data. The NCBI has also designed user-friendly ways to access and analyze an incredible amount of data on nucleotide sequences, protein sequences, molecular structures, genome data, and even scientific literature. DNA database searching. Try it yourself. For example, an NCBI program called Basic Local Alignment Search Tool, BLAST, www.ncbi.nlm.nih.gov/blast can be used to search GenBank for sequence matches between cloned genes and to create DNA sequence alignments. Go to the BLAST website and click Standard Nucleotide BLAST done. In the search box, type in the following sequence, EATAGA. Imagine that this sequence is from a piece of a gene that you just cloned and sequenced and you want to know if anyone cloned this gene before you. Click the BLAST button. Your results will be available in a minute or two. Click the Format button to see the results of your search. A page will appear with the results of your search. You may need to scroll down the page to find the sequence alignment. What did you find? Figure 3.21 shows a BLAST search alignment comparing the human and mouse gene sequences for an obesity gene, OB, that produces a hormone called leptin. Leptin plays a role in fat metabolism, and mutations in the leptin gene can contribute to obesity. See also Figure 11.1. Notice that the nucleotide sequence for these two genes is very similar, as indicated by the vertical lines between identical nucleotides. The Human Genome Nomenclature Committee, supported by the NIH, establishes rules for assigning names and symbols to newly cloned human genes. Each entry into GenBank is provided with an accession number that scientists can use to refer back to that clone sequence. For example, go to the GenBank website listed at the companion website and then type in the accession number U14680 and click Go. What gene is identified by this accession number? Click on the accession number link. Notice that GenBank provides the original journal reference that reported this sequence, the single-letter amino acid code of the protein encoded by this gene, and the nucleotide sequence, cDNA, of this gene. Alternatively, you can type in the name of a gene or a potential gene that you are. Figure 3.21 Comparison of the human and mouse OB genes. Partial sequences for these two genes are shown, with the human gene sequence on top and the mouse gene sequence below it. Interested in to see if it has already been cloned and submitted to GenBank. The BLAST search will identify a match with a human gene for early onset breast cancer, BRCA1. The GenBank search identifies the same gene by its accession number. GenBank is only one example of an invaluable database that is an essential for bioinformatics. Many other specialized databases exist with information such as single nucleotide polymorphism data, BAC, and YAC library databases, and protein databases that catalog amino acid sequences and three-dimensional protein structures. A genome cloning effort of epic proportion, the Human Genome Project. It is a very exciting time to be studying biotechnology. We are fortunate to witness the completion of an unprecedented project that involved many of the techniques described in this chapter, the Human Genome Project, HGP. Initiated in 1990 by the U.S. Department of Energy, DOE, the HGP was an international collaborative effort with a 15-year plan to identify all human genes, originally estimated at 80,000 to 100,000 in number, and to sequence the approximately 3 billion base pairs thought to make up the 24 different human chromosomes, chromosomes 1 to 22, X, Y. The HGP was also designed to accomplish the following. Analyze genetic variations among humans. This included the identification of single nucleotide polymorphisms, SNPs, see Figure 1.11. Map and sequence the genomes of model organisms, including bacteria, yeast, roundworms, fruit flies, mice, and others. Develop new laboratory technologies such as high-powered automated sequencers and computing technologies, as well as widely available databases of genome information, which can be used to advance our analysis and understanding of gene structure and function. Disseminate genome information among scientists and the general public. Consider the ethical, legal, and social issues that accompany the HGP in genetic research. In the United States, public research on the HGP was coordinated by the National Center of Human Genome Research, a division of the NIH and the DOE. The project funded 7 major sequencing centers in the United States. Over time, the project grew to become an international effort with contributions from scientists in 18 countries. The work was primarily carried out by the International Human Genome Sequence Consortium, which involved nearly 3,000 scientists working at 20 centers in 6 countries: China, France, Germany, Great Britain, Japan, and the United States. The estimated budget for completing the genome was $3 billion, a cost of $1 per nucleotide. Driven in part by competition from private companies in the development of computer-automated DNA sequences, such as the one described in Figure 3.12, and bioinformatics, the HGP turned out to be a rare government project that completed all of its initial goals and several additional goals more than 2 years ahead of schedule and under budget. The most aggressive competitor on the project was a private company called Celera Genomics, aptly named from a Latin word meaning swiftness, directed by Dr. J. Craig Venter, who was the scientist directing the Institute for Genomic Research when it used shotgun cloning to sequence the genome for H. influenza, as discussed earlier in this section. Celera announced its intention to use its novel shotgun sequencing approach, similar to the methods they had used to sequence the genome for H. influenza, as well as newly developed, high-powered computer-automated DNA sequencers, to sequence the entire human genome in 3 years. Fearful of how private corporations might control the release of genome information, U.S. government groups involved in the HGP were effectively forced to keep pace with private groups to stay competitive in the race to complete the genome. In 1998, as a result of accelerated progress, a revised target date of 2003 was set for completion of the project. On June 26, 2000, leaders of the HGP and Celera Genomics participated in a press conference with President Bill Clinton to announce that a rough working draft of approximately 95% of the human genome had been assembled, nearly 4 years ahead of the initially projected timetable. At a joint press conference on February 12, 2001, Dr. Francis Collins, director of the NIH National Human Genome Research Institute and of the HGP, and Dr. J. Craig Venter of Celera announced that a series of papers describing the initial analysis of the genome working draft sequence were published by their research groups in the prestigious journals Nature and Science, respectively. Scientists spent the next 2 years working to fill in thousands of gaps in the genome by completing the sequencing of pieces not yet finished, correcting misaligned pieces, and comparing sequences to ensure the accuracy of the genome. On April 14, 2003, the International Human Genome Sequencing Consortium announced that its work was done. A map of the human genome was essentially complete, with virtually all bases identified and placed in their proper order and potential genes assigned to a chromosome. See Figure 3.22. What have we learned from the human genome? One of the most surprising findings of the project was that the human genome consists of approximately 20,000 protein-coding genes, not 100,000 genes as predicted. The prediction was based primarily on estimates that human cells make approximately 100,000 to 150,000 proteins. One reason why the actual number of genes is so much lower than the predicted number is the discovery of large numbers of gene families with related functions. In addition, many genes that code for multiple proteins through alternative splicing have been found. It has been estimated that 92% to 95% of human genes produce multiple proteins through alternative splicing. Analysis of human genes by functional categories has provided genome scientists with a snapshot of the numbers of genes involved in different molecular functions. Figure 3.23 shows one proposed interpretation of assigned functions to human genes based on gene sequence similarity to genes of known function. Not surprisingly, many genes encode enzymes, while other large categories of genes encode proteins involved in signaling and communication within and between cells and DNA and RNA binding proteins. Notice that this estimate also shows that approximately 42% of human genes have no known function, although recent evidence suggests that functions for over half of our genes remain unknown. Keep this in mind if you are interested in a career in genetics research. because efforts to discover what these genes do will provide exciting career opportunities for many years into the future. Figure 3.22 estimated number of genes on each chromosomes and approximate size in base pairs, BP, of each human chromosome. Figure compiling statistics for human chromosomes are based on the Sanger Institute's Human Genome Information in the Vertebrate Genome Annotation, VEGA, database. Number of genes is an estimate, as it is in part based on gene predictions, which is why this value is higher than the estimated 20,000 genes generally accepted as the number of human genes. Total chromosome length is an estimate as well, based on the estimated size of some non-coding sections of the genome that remain unsequenced. Figure 3.23 proposed functions for the numbers of human genes assigned to different functional categories. Parentheses show percentages based on the 2001 published data by Venter et al. For 26,383 genes. Here is a summary of important highlights of some of the basic concepts scientists have learned from the HGP. The human genome consists of approximately 3.1 billion base pairs. The genome is approximately 99.9% the same between individuals of all nationalities. Single nucleotide polymorphisms, SNPs, and copy number variations, CNVs, such as long deletions, insertions and duplications in the genome account for much of the genome diversity identified between humans. Less than 2% of the genome codes for genes. The vast majority of our DNA is non-protein coding, and repetitive DNA sequences account for at least 50% of the non-coding DNA. The genome contains approximately 20,000 protein-coding genes. Many human genes are capable of making more than one protein, allowing human cells to make at least 100,000 proteins from only about 20,000 genes. The functions of over half of all human genes are unknown. Chromosome 1 contains the highest number of genes. The Y chromosome contains the fewest genes. Many of the genes in the human genome show a high degree of sequence similarity to genes in other organisms. Thousands of human disease genes have been identified and mapped to their chromosomal locations. How will we benefit from the HGP? The complete sequence of the human genome has been described as the genetic blueprint of humanity, containing the scientific keys to understanding our biology and behaviors. Identifying all human genes is not as important as understanding what these genes do and how they function. We will not fully understand the function of all human genes for many years, if ever. One immediate impact of the HGP will be the identification of genes associated with human genetic diseases. We will discuss some of the ways the HGP is changing diagnosis and treatment of genetic diseases, Chapter 11. A better understanding of the genetic basis of many diseases will lead to the development of new strategies for disease detection and innovative therapies and cures. For updated information on the HGP and for an outstanding overview of goals, sequencing, and mapping technologies, and the ethical, legal, and social issues of the HGP, visit the Human Genome sites listed at the companion website. The Human Genome Project started an omics revolution. The HGP and genomics are largely responsible for ushering in a new area of biological research, the omics. It seems that every year, new or existing areas of biological research are being described as having an omics connection. For example, proteomics studying all of the proteins in a cell. Metabolomics studying proteins and enzymatic pathways involved in cell metabolism. Metabonomics measuring metabolic products produced by cells in response to stimuli, such as drug treatment, and genetic manipulation. Glycomics studying the carbohydrates of a cell. Transcriptomics studying all genes expressed, transcription, in a cell. Metagenomics the analysis of genomes of organisms collected from the environment. Pharmacogenomics customized medicine based on a person's genetic profile for a particular condition. If any of the omics areas defined here sound of interest to you, do some internet research on these topics and you will find vast amounts of information about each area. We will touch on many of these omics areas throughout the book. As further evidence of the impact of genomics, a new field of nutritional science, called nutritional genomics or nutrigenomics, has emerged. Nutrigenomics is focused on understanding interactions between diet and genes. Several companies provide nutrigenomics tests, using microarrays or other genetic tests to analyze your genotype for genes thought to be associated with different medical conditions or aspects of nutrient metabolism. These companies then provide a customized report on nutrition and claim that they can suggest dietary changes you should make to improve your health and prevent illness based on your genes. Whether nutrigenomics is actually a valid scientific approach is a subject of debate among scientists. Comparative genomics. It might surprise you that in addition to studying the human genome, the HGP involved mapping and sequencing genomes from a number of model organisms, including E. coli, a model plant called Arabidopsis thaliana, the yeast Saccharomyces cerevisiae, the fruit fly Drosophila melanogaster, the nematode roundworm Sinorhabditis elegans, and the mouse Mus musculus, among other species. Complete genome sequences of these model organisms have been incredibly useful for comparative genomic studies, which allow researchers to study gene structure and function in these organisms in ways designed to understand gene structure and function in other species, including humans. Because we share many of the same genes as flies, roundworms, and mice, such studies will also lead to a greater understanding of human evolution. The number of genes we share with other species is very high, ranging from about 30% of the genes in yeast to about 80% of the genes in mice and about 95% of the genes in chimpanzees. Table 3.3. Comparative genomic analysis of the genome for man's best friend has revealed that we share about 75% of our genes with dogs. Human DNA even contains around 100 genes that are also present in many bacteria. When researchers completed the 814 million BP genome of the sea urchin, Strongylocentris purpuratus, an invertebrate that has served as an important model organism particularly for developmental biologists, we learned that of the 23,500 genes in the urchin genome, many are genes with important functions in humans. The genomes of many model organisms have been completely sequenced, and there are literally hundreds of genomics projects underway worldwide. Genome scientists around the world have proposed to sequence 10,000 vertebrate genomes, the Genome 10K plan. This ambitious plan proposes to assemble 10,000 genomes in 5 years, about one genome a day. The first genome for a tree, the black cottonwood, a type of poplar, was sequenced several years ago. To date, the poplar's 45,555 genes are the highest number found in a genome. Scientists anticipate using data from this project to help the forestry industry make better products, including biofuels or even genetically engineered poplars to capture high levels of carbon dioxide from the atmosphere. The honeybee genome has also been completed, and information from this project will be used to understand bee genetics and behavior to help the honey-producing industry as well as advance an understanding of how bee toxins produce allergic responses. We will discuss genomics projects in other chapters of this book, and throughout it you will learn about many applications of genomics. For example, in Chapter 5 we discuss microbial genome sequencing projects that are sequencing genomes for literally hundreds of newly identified microbes, including microbes that live in on humans, the Human Microbiome Project, and microbes present in water, soil, and air samples from around the world, metagenomics. We will also discuss synthetic genome approaches designed to create novel life forms from artificially constructed genomes. Stone Age genomics. As yet another intriguing example of genomics, a number of labs around the world are involved in analyzing ancient DNA. These studies are generating fascinating. Table 3.3 Comparison of selected genomes. Organism, scientific name. Approximate size of genome, date completed. Number of genes. Approximate percentage of genes shared with humans. Web access to genome databases. Bacterium, Escherichia coli. 4.1 million BP, 1997. 4403. Not determined. www.genome.wisc.edu. Chicken, Gallus gallus. 1 billion BP, 2004. 20,000,23,000. 60. http dog, Canis familiaris. 6. 8. HT Genome. Data from minuscule amounts of ancient DNA from bone and other tissues and fossil samples that are tens of thousands of years old. Analysis of DNA from a 2,400-year-old Egyptian mummy, mammoths, platypuses, Pleistocene age cave bears, and Neanderthals are some of the most prominent examples of Stone Age genomics, also called paleogenomics. Researchers from McMaster University in Canada and Pennsylvania State University published partial sequence data, about 13 million BP, from a 27,000-year-old woolly mammoth. This study showed that there is approximately 98.5% sequence identity between mammoths and African elephants. Subsequent work by other scientists has used whole genome shotgun sequencing of mitochondrial and nuclear DNA from Siberian mammoths to provide data on the mammoth genome. These studies suggest that the mammoth genome differs from that of the African elephant by as little as 0.6%. These studies are a great demonstration of how stable DNA can be under the right conditions, particularly when frozen. Also intriguing are similarities that have been revealed between the mammoth and human genomes. When the gene sequences from human chromosomes were aligned with sequences from the mammoth genome, approximately 50% of mammoth genes show sequence alignment with human genes on autosomes. In 2009, a team of scientists led by Svante Paabo at the Max Planck Institute for Evolutionary Anthropology in Germany and 454 Life Sciences reported completion of a rough draft of the Neanderthal, Homo neanderthalensis, genome encompassing more than 3 billion BP of Neanderthal DNA and about two-thirds of the genome. In 2006, Paabo's group, along with a number of scientists in the United States, reported the first sequence of about 65,000 BP of nuclear DNA isolated from bone of a 38,000-year-old Neanderthal sample from Croatia. Because Neanderthals are close relatives of humans, the sequencing of the Neanderthal genome provides a tremendous opportunity to use comparative genomics to advance our understanding of evolutionary relationships between humans. The human and Neanderthal genomes are 99% identical. Some of these sequences are involved in cognitive development and sperm motility. Of the many genes shared by these species, FOXP2 is a gene which has been linked to speech and language ability. There are many genes that influence speech, so this finding does not mean that Neanderthals spoke as we do. But because Neanderthal had the same modern human FOXP2 gene, scientists have speculated that Neanderthals possess linguistic abilities. The realization that modern human and Neanderthals lived in overlapping ranges as recently as 30,000 years ago has led to speculation about the interactions between modern humans and Neanderthals. Genome studies suggest that there was interbreeding between Neanderthals and modern humans an estimated 45,000 to 80,000 years ago in the eastern Mediterranean. These exciting studies, previously thought to be impossible, are having ramifications in many areas of human evolution, and it will be interesting indeed to follow the progress of this work. 10 years after the HGP, what is next? In the 10 years since completion of a draft sequence of the human genome, studies on the human genome continue at a very rapid pace. As a result of the HGP, many other major theme areas for human genome research have emerged, including cancer genome projects, analysis of the epigenome, including the Human Epigenome Project, which is creating hundreds of maps of epigenetic changes in different cell and tissue types and evaluating potential roles of epigenetics in complex diseases, characterization of SNPs, the International HapMap Project, and CNVs for their role in genome variation, disease, and pharmacogenomics. Applications. As the HGP was being completed, a group of about 36 research teams around the world began the Encyclopedia of DNA Elements (ENCODE) project. The main goal of ENCODE is to use both experimental approaches and bioinformatics to identify and analyze functional elements, such as transcriptional start sites, promoters, and enhancers, that regulate expression of human genes. Here we discuss two major human genome-related research project areas: personalized genome projects and cancer genome projects. Personalized genome projects. Because of rapid advances in sequencing technologies, several companies have even proposed to sequence genomes for individual people, or personalized genomics. In 2006, the X Prize Foundation announced the Archon X Prize for Genomics, a project to award $10 million to the first group to develop technology capable of sequencing 100 human genomes with a high degree of accuracy in 10 days for under $10,000 per genome. Other groups are working on sequencing a personalized genome for a mere $1,000. Pursuit of the $1,000 genome is evidence that DNA sequencing may eventually be affordable enough for individuals to consider acquiring a readout of their own genetic blueprint. In 2007, the company 454 Life Sciences together with researchers at Baylor University sequenced James Watson's genome for approximately $1 million. 454 Life Sciences decided that Project GEM, sequencing the genome of the Cody's cover of the DNA structure, was a good high-profile way to develop and promote their new next-generation sequencing technology. James Watson provided 454 scientists with a blood sample in 2005 and, in mid-2007, they presented Dr. Watson with 2 DVDs containing his genome sequence, which was completed at a rough cost of just under $1 million. Watson has allowed his sequence to be available to researchers except for the sequence of his apolipoprotein E gene, APO. APO gene mutations can indicate a disposition for Alzheimer's disease. Human genome pioneer Craig Venter also had his genome sequenced by scientists at the Craig J. Venter Institute. George Church and colleagues at Harvard University have started the Personal Genome Project, PGP, and have recruited volunteers to provide DNA for individual genome sequencing with the understanding that the resulting genome data will be made publicly available. Church's genome has been completed and been made available online. Since the Watson and Venter genomes were completed, the first complete genome sequence was provided for an individual ancient human, a Paleo-Eskimo, obtained from a roughly 4,000-year-old permafrost-preserved hare. This work recovered about 78% of the diploid genome and revealed many interesting SNPs, of which about 7% have not been previously reported. You decide to sequence or not to sequence. As of early 2010, 13 individual human genome sequences have been reported, including sequences for a Yoruba African person, 2 individuals of Northwest Europe origin, a Han Chinese individual, 2 persons from Korea, African Archbishop Desmond Tutu, a family of 4, and several others. Would you want your genome sequenced? What would you pay for your genome sequence? What would you do with this sequence information? Who would have access to your sequence information? You decide. Career profile: career options in the biopharmaceutical industry, perspectives from a recent graduate. Biopharmaceutical companies discover new drug treatments and develop them from the research and development stages through to commercial manufacturing, marketing, and finally sales. What makes the biopharmaceutical industry or biopharma different from the traditional pharmaceutical industry? Pharmaceutical companies in general develop chemically based drug compounds that treat illness non-specifically, whereas biopharma uses knowledge of biological systems to develop biologically derived drug compounds that treat illness in a highly specific manner. Although biopharma is currently much smaller than the pharmaceutical industry, its growth is triggered by the increasing awareness of the potential to treat illnesses effectively and possibly to find cures for illnesses that were once thought incurable. The lines between traditional pharma companies and biopharma are blurred. Many large pharma companies have biotechnology divisions that work on biopharma. For example, the pharma company Roche recently purchased Genentech. Career paths within the biopharmaceutical industry are similar to those in the pharmaceutical industry, owing to the similarity in the path of drug development. The career possibilities are wide-ranging, however, gaining employment with a biopharmaceutical company is not easy. Competition is fierce, and the truth is that no matter how well a person may fit a position, there is always someone else who will be just as qualified. For that reason, start thinking about career options shortly after you have selected your major. The best way to begin is by becoming familiar with the industry. Many of the websites cited on the companion website are excellent resources for learning about different job opportunities. This knowledge will enable you to customize your education to better serve a particular job function, a sure way to get ahead of the competition. Many entry-level positions are available to those with associate's and bachelor's degrees. However, having a bachelor's degree will guarantee a slightly higher starting salary. Many higher-paying entry-level positions require an advanced degree, such as a master's degree or even a doctorate, PhD. Knowing this may greatly influence your decision to continue your education before entering the workforce. However, as important as educational training is, employers are constantly looking for experienced individuals. Gaining real-life experience through volunteer work, independent study research, or an internship may offer you the best opportunity to explore your options while you are completing your studies. Industry internships are an excellent way to learn about different roles in a company and to see what you like, and they may also lead to employment after graduation. If you cannot find the ideal job in the industry after graduation, temporary employment is another good way to begin with a biopharmaceutical company. Many placement services work with job seekers and biotechnology companies to find people who can fill a need for a short period of time. However, if you do a good job in a temporary position, the company may try to find a permanent position for you. In considering a position, it is important to understand the job description as well as what is required of that position. Numerous companies hire people as lab technicians, but the job descriptions for this position are as numerous as the jobs. For example, quality control lab technicians follow very controlled and highly precise procedures to test the quality of a product. Their main function is to ensure that a good product is being produced. Technicians working in a diagnostic capacity are responsible for testing a new product or procedure to make sure that it works correctly and then for recommending improvements. Research and development or discovery lab technicians are responsible for just that. Their main function is to develop new products as well as to find new uses for old products. It is not required that you have prior experience in every experimental assay or lab procedure to become a lab technician. A good company will always teach you what you need to know to do the specific job. However, all lab technicians must have good lab skills and be organized, detail-oriented, motivated, and, most importantly, enthusiastic. A potential downside of working in the biopharmaceutical industry is that all work is tailored around developing, producing, and selling a product. Creativity and originality are not always incorporated into the daily routine of life in the laboratory. The work environment is usually very controlled because all operations are regulated by the FDA and must be documented rigorously. Also, since products are produced through biological processes, work schedules are linked directly to these processes, such as waiting for cells to grow to the right density. In some cases, this can mean a shorter work week or less structured work hours. In the biopharmaceutical industry, the fast-paced work environment is always changing and always demanding that its employees work hard and efficiently. Each day brings new obstacles and challenges, which makes working in this industry so exciting. Contributed by Robert Sexton, B.S. Biology, MBA, Monmouth University, Oncology Development Operations, Novartis Pharmaceuticals Corporation. Cancer Genome Projects. Cancer as a disease has many genetic components. The NIH began work on a cancer genome project called the Cancer Genome Atlas Project, TCGA, to map important genes and genetic changes involved in cancer. The TCGA project has sequenced over 100 partial genomes for various cancers to date. Together with a group of researchers in 11 countries, called the International Cancer Genome Consortium, JCGC, there are plans to sequence genomes from over 500 tumor samples representing more than 20 different cancers. Not surprisingly, most common cancers such as breast, lung, colon, pancreatic, brain, and ovarian cancer are among the most actively studied cancer types and are being analyzed first. Ultimately, it is expected that the identification of key genes involved in tumor formation and metastasis, spreading, will lead to improved diagnostic techniques for the detection of cancer and more effective treatments for curing cancer. This is already happening in some cases, as we will discuss in Chapter 11. Making a difference. One of the most successful examples of how recombinant DNA technology is saving lives is also one of its first applications for treating human disease. Earlier in the chapter we discussed how the recombinant form of insulin, called Humulin, became the first product of recombinant DNA technology to be approved by the FDA for use in humans. Prior to recombinant insulin, insulin was isolated from animals. In many diabetics, insulin from animal sources was ineffective and caused a range of complications. Producing recombinant insulin was not easy, see Figure 5.9. Insulin is composed of two different polypeptide chains. Both polypeptides had to be expressed in recombinant form and assembled to form the active hormone. Needless to say, it was quite a feat to successfully engineer insulin, such a challenging recombinant protein, as the first product of this technology. This demonstration that a recombinant approach for producing a complex protein was possible made scientists highly optimistic about the possibilities of recombinant DNA approaches to making other proteins of therapeutic value. Humulin was originally developed by Genentech and then widely distributed by Eli Lilly and Company, which continues to produce Humulin today. Since it first became available in 1982, Humulin has been used successfully and safely to treat millions of patients. The vast majority of insulin given to insulin-dependent diabetes mellitus patients worldwide is recombinant insulin. There are now many variations of Humulin available, most of which are different mixtures that determine whether the insulin is fast or slow-acting once injected. Recently, researchers have been working on techniques to produce recombinant insulin in plants, which is expected to dramatically reduce biomanufacturing costs. The insulin story is an outstanding example of how recombinant DNA techniques are being used to make a difference in alleviating human pain and suffering.
And that completes the entire Chapter 3. It was about 40 pages read, and it completes this 2-hour, 37-minute Camtasia.